welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event.
Welcome to our Day 2 of the FDA Clinical Investigator Training Course 2023. Good day, good night, or any time in between from wherever you may be worldwide. My name is Ray Ford, and I'm with the Small Business and Industry Assistance Program, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research's Office of Communication. It's my pleasure, along with my CEDAR colleagues, to be with you today. We ask you to make the most of this experience and once again submit your questions during the available Q&A session panels. We're also offering live YouTube streaming, so invite your colleagues. One thing I need to clarify, you must register and attend live to collect CE. CE credit is not available for watching the recordings. Also, please remind you that this is day 2 of the workshop, offering a total of 9.75 contact hours to physicians, pharmacists, and nurses. The CEs are available to all real-time attendees who self-identified as healthcare professionals during the registration process. Once registered, you must attend live in real time to collect CEs. CE credit is not available for watching the recordings. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than that of a single supporting company. CE faculty and speakers are required to disclose to the attendees when the products or procedures being discussed are off-labeled or unlabeled, not FDA approved, and any limitations on the information that is presented. We have a spectacular lineup of speakers once again, and all relevant financial relationships listed for these individuals have been mitigated. The planning committee, speakers, and CE consultation and accreditation team have nothing to disclose. Participants must attest to their attendance and complete the final activity evaluation via the CE portal. The web address is ceportal.fda.gov. For multi-day activities, participants must attest to their attendance and complete the faculty evaluation for each day. Final activity evaluations must be completed within 2 weeks after the activity, no exceptions. So in this case, the deadline for this 2-day course is December the 21st. I apologize in advance, but we are unable to make exceptions to that deadline. Pharmacists should note that failure to provide your correct NABP and date of birth information in the required format may result in loss of credit for this activity. The NABP profile number should be the 6 to 7-digit profile number assigned by the CPE monitor, and your birth date should be in the month, day format, such as 1207. Do not provide your pharmacy license number. Once you log in, please click the My Account tab, then navigate to the Edit Contact Information to verify that your information is correct. Physicians and nurses may then view and print a statement of credit. Pharmacists should log into the CPE monitor 8 weeks after the last session of the activity to obtain their CE credit. To simplify things, each night, SBIA will send one email to the attendees of that individual event day. The email will have information on both the CE claim codes and the survey link. This slide provides QR codes for our social media avenues. You can scan them now with your phones and be taken to the appropriate platform. They'll also be advertised during the breaks if you miss it now. Turning to logistics and our ongoing environment, this event is 100% remote and virtual, and as such, there may be some internet issues, and if we encounter those, we'll try to get them resolved as quickly as possible. Speaking of tech issues, here's a link for our one-stop shop for answers to your technical questions, downloading files, accessing the survey link to obtain the certificate of attendance, and much more. That's the same page you used this morning for your online access to the event, so please bookmark that page. We invite you to submit your questions so that we have a vibrant and productive QA panel. When you submit your question, please identify the individual presenter by prefacing your question with the speaker's name as shown here at Prisma. Also, during each presentation, there's a link to the right side of the pod where at the speaker's headshot, it says download the slides. Note, this link is only active during the real-time presentation. Also, if you need closed captioning, please look at the top of your your screen to the right. Click on the CC drop-down menu and select Show Captions. There will be a link to the speaker evaluation under the speaker's picture or information on the right side of the screen. We encourage you to take time also to complete that evaluation, as your feedback is much appreciated. In addition to the speaker evaluation, we remind you of our final evaluation. The final survey is a mechanism by which you receive the certificate of attendance. When an attendee completes a survey evaluation, they can then download a certificate of attendance which may be used in support of RAP, SOCRA, ACRP, and SQA credits. The certificate must be downloaded immediately after completing the survey. Otherwise, this opportunity is lost. Even if you do not want or need the certificate, please take a few minutes to complete the conference evaluation to help guide the design of our future events. Note that the evaluation and the certificate are only available for 2 weeks, and that takes us to the deadline of December 21st. The survey link will be posted on our one-stop shop page this afternoon after the event. All the sessions are being recorded. It'll take us a week or so to process recordings, but you'll soon be able to access them through SBI's webpage, and that's fda.gov/CEDAR-SBIA. In the interim, the YouTube link for each day's recording will be posted on our one-stop shop webpage at the end of the day. Please visit our CEDAR Learn webpage, where you can link to SBIA tutorials along with other educational resources, many of which offer CEs, and that's fda.gov/CEDAR Learn. Let me remind you of a few of our upcoming events on December the 12th and the 13th. Join us for the webinars on navigating complex orders, a deep dive into FDA drug interactions, guidances, and resources. January the 18th, also join us for FDA's Model Integrated Industry Meetings Pilot Program for Generic Drugs webinar. And on February the 13th through the 15th, 2024, we're hosting a joint US FDA, MHRA UK Health Canada Good Clinical Practice and Pharmacovigilance Compliance Workshop. For details on each event, please see our website at fda.gov/CEDAR-SBIA. Now, we do have our special announcement we'd like to bring to your attention once again to make on enhancing the adoption of innovative clinical trial approaches to understand the state of innovation in clinical trial design and conduct. FDA is asking for perspectives via comments to our public document, and that's FDA-2023-N-4489 by April the 19th, 2024, and or participation in the public workshop hosted in partnership with the Duke Margolis Center for Health Policy. That'll be on March the 19th and 20th, 2024. For more information, you can also please contact Dr. Kevin Bugan, listed here at the provided email address. There's also QR codes which you can access this more information. We'd like to welcome back Dr. Leonard Sachs to provide us with some opening remarks to prepare us for today's presentations. It's my pleasure to present Dr. Sachs.
Thank you very much, Ray. And good morning to everybody. Welcome back to day 2 of our clinical investigator training course. So yesterday, those who were present know that we focused on clinical trials. We focused on their design, some of the opportunities and challenges, specific populations that were challenging, rare diseases. We also spoke about the analysis of clinical trials. And this morning, we will turn to non-clinical and early clinical data that are critical before proceeding with a clinical trial. Is the quality of the investigational product adequate? Do we have enough preclinical or toxicology data to ensure safety in humans? And finally, do we understand how the body handles the drug? So to address these issues, I'm delighted to welcome Pinky Patel from the Office of Product Quality to tell us about chemistry and manufacturing controls. She'll be followed by Matthew Thompson from the Office of New Drugs with a discussion on pharmacology and toxicology. And finally, Shirley South from the Office of Translational Science will review the clinical pharmacology data that we need. So without any further ado, let's turn it over to Pinky to begin.
Good afternoon. My name is Pareshma Patel, and I'm currently a Division Director within CDER's Office of Pharmaceutical Quality. Today, I'll be talking about chemistry, manufacturing, and controls, and some regulatory considerations through clinical development, with a focus on small molecule CMC development. By the end of this presentation today, you should be able to understand the regulatory definitions and requirements for drug substances and drug products, describe chemistry, manufacturing, and controls information for IND submissions, and name some potential CMC safety concerns for INDs. Today, I'll start off with a brief definition of pharmaceutical quality, and then go through how chemistry, manufacturing, and controls changes through clinical development, from the submission of an IND through submission of a marketing application or an NDA. I'll provide some key regulatory definitions and CMC requirements that are outlined in the Code of Federal Regulations and further described within guidance documents for INDs that are published by the FDA. I'll discuss some specific requirements for drug substance and drug products and the key CMC information that's submitted for these two sections of INDs. I'll provide a brief description of the main issues related to CMC that are considered IND safety concerns and could lead to clinical holds. And I'll end with some key guidance documents and resources as references. I'd like to start with an overall view of the mission and vision of the Office of Pharmaceutical Quality. Everyone deserves confidence in their next dose of medicine, and pharmaceutical quality assures the availability, safety, and efficacy of every dose. This is essentially the end goal when moving from clinical development to commercialization. One challenge to the manufacturing aspects of an application is that it covers the life cycle of a product, and it often rapidly changes from an IND stage of development to commercialization. From pre-IND or preclinical stages to IND submission, there may be very limited CMC information. And as the product development proceeds through pivotal clinical studies, we expect significant CMC development to assure consistent manufacturability to support a marketing application or an NDA. Even following an NDA approval, the CMC information can still change significantly as suppliers or manufacturers may change, and keeping in mind that pharmaceutical manufacturing is a global business with complicated supply chains. Throughout this process, the CMC or product quality team is meeting with sponsors to advise them on key aspects of CMC development as they move closer to commercialization. The key challenge for the quality review is by the time you get to a marketing application, we want to assure that the characteristics and performance of the drug that was used in the pivotal clinical studies will be consistently replicated in the commercially marketed batches, while assuring an adequate supply can also be maintained. I would like to start by providing two key regulatory definitions for drug substance and drug product. These are defined in the Code of Federal Regulations, and this is generally the framework and organization of information that is submitted to both INDs and marketing applications. The drug substance is an active ingredient that's intended to furnish the pharmacological activity or other direct effect in the diagnosis, cure, mitigation, treatment, or prevention of a disease, or to affect the structure or function of the human body. The drug product is the finished dosage form, and it contains the drug substance and may or may not contain other ingredients. The requirements for CMC information to be submitted in an IND are described in the CFR, and this is now electronically available. The main text of the CFR can be broken down into a few main requirements for CMC information for an IND. They include the composition and description of how the drug substance and drug product are controlled, information on the manufacturer, so who is making the drug, how they make it, which is essentially the manufacturing process, information to assure identity, quality, purity, and strength of the investigational drug, and then demonstration that the drug will be stable during its clinical use. And it's really important to note that the regulations give FDA flexibility for the amount of data that should be submitted, depending on the phase of investigation, the dosage form, the duration of use, and any other available information. The regulations also specifically emphasize that for a phase 1 submission, the emphasis is really on the identification and controls of the materials used to make the drug and the new drug substance itself that's under investigation. I would also like to point out that according to the regulations, FDA has 30 days to review these submissions, and if sponsors do not hear back from the agency, that they may initiate clinical studies. So significant FDA resources go into these types of IND reviews. I would like to also take a minute to highlight that the IND assessment teams are multidisciplinary in nature. The teams include members such as clinical reviewers, clinical pharmacology, statistics, pharmacology, toxicology, in addition to potential consults to other centers and other disciplines as needed. During the IND review for an original IND or first-in-human phase 1 studies, there typically may only be 1 to 2 CMC or product quality reviewers on this team, depending on the specific product being reviewed. However, as development proceeds beyond those early stages of development, the product quality team expands to include subdiscipline expertise. So by the time a marketing application is submitted, the product quality team includes members from the following disciplines: drug substance, drug product, biopharmaceutics, which really considers in vitro dissolution of the final dosage form, drug product manufacturing, evaluation of facilities, microbiology, and finally surveillance and inspections that may be needed for the facilities that manufacture the drug substance or the drug product. Today, I'm going to focus mostly on the drug substance or active pharmaceutical ingredient information that FDA expects as part of an IND application during review. I'm focusing on drug substance because, as stated in our regulations, the emphasis for phase 1 is really the safety of that drug substance. I'll briefly discuss some of the general requirements of what the FDA is looking for as part of the review of INDs. First is general information, which includes the structure, molecular weight, formula, and the source of the drug substance. The manufacturing process is essentially the chemistry for how a drug substance is made. Then I'll move to characterization data, which I'll cover in 2 parts: structural characterization data and characterization of the physical chemical properties of the drug substance. As part of the characterization section, there's also information about the understanding of impurities and the sources of impurities that are generated as part of the drug substance manufacturing process. I'll briefly touch on controls, part of CMC, which really defines the release specification that are used in order to release batches of drug substance for use in clinical studies. Batch data includes the actual data for the batches intended for use in the clinical studies. And finally, data to support stability for the drug substance and drug product intended for use in the clinical studies. One key aspect of our regulations is that they are broadly applicable to APIs with significantly variable structures. As part of regulatory submissions, information on the source of drug substance is submitted, and there are a wide variety of APIs that are regulated as drugs that we review in our office. There are drugs manufactured using chemical synthesis, which includes small molecules. Shown here are just a couple of examples: Alaxacaftor and nirmatrelvir. Additionally, peptides can also be manufactured using chemical means, such as solid-phase peptide synthesis. Shown here is just an example of plecanatide. And then oligonucleotides, such as siRNAs or antisense oligonucleotides, also fall under this category and can be manufactured using chemical synthesis. Shown here are just the 2 examples: patacerin sodium as well as tofersen. There are also drug substances derived from fermentation products, such as erythromycin, or semi-synthetic drugs. Shown here as an example is the example of paclitaxel, where the portion in red is isolated from the yew tree, and chemical synthesis is used to modify the structure further. There are also a wide variety of drugs that are isolated from natural sources, including drugs from inorganic sources, botanical sources like the synecatechins shown here, and animal sources such as heparin, which is derived exclusively from the intestinal mucosa of pigs. And we also review combination products such as antibody-drug conjugates in collaboration with the Office of Biotechnology Products, with our focus being on the chemistry and conjugation of the payload and linker to the antibody. Hopefully the last couple of slides gave you an idea of the molecular variability and science that chemists within FDA have to understand in order to review and regulate products. In addition to the information related to the actual structure and source of the drug substance, the bulk of the review for drug substance is really the chemistry for how the drug substance is made. I'll focus on new molecular entities today because the manufacturing process is typically at an early stage of development when an IND is submitted. In order for us to assess the safety of the drug substance to be used in clinical studies, we ask for a brief description of the synthesis used to manufacture the clinical trial material. This information should be provided in both a written format as well as a synthetic scheme that provides us with a complete picture of all the chemical reagents, solvents, catalysts that are used as part of the manufacturing process, and information on any of the controls for the purity and quality of these materials. In addition, as part of the manufacturing section, we often get information on any preliminary and process controls, which are really the analytical test to ensure reaction completion as part of this at each step of a process. For our first in-human NME IND, the emphasis really is on the difference in the quality of toxicology batches that were used for non-clinical studies and the clinical batches planned to be used. And we work closely with our pharmacology and toxicology colleagues to assess any differences between these manufacturing processes or the quality of these batches. I would also like to just emphasize that FDA does not expect that all of this information is fully understood at the time of IND submission. We're aware that the manufacturing process is being optimized for scale, yields, purity, and to minimize use of toxic or dangerous chemicals. Additionally, we're also aware that minimal steps are usually conducted under GMP at these early stages of development, and the commercial GMP manufacturing process has not been developed or finalized quite yet. FDA does encourage that sponsors initiate a CMC-focused meeting prior to initiation of pivotal clinical studies to really address any CMC challenges or to come to agreement on any specific CMC aspects related to manufacturing of an API. Next, I'm going to move on to talk about the characterization that's expected for a drug substance. I'll group this into two categories: structural characterization data and physical chemical characterization data. First is the structural characterization data. This is the data to support the proposed structure of the drug substance itself. And we recognize that structural characterization of the drug substance may be limited early in development, but as part of this characterization, we need enough data to assure identity, and we want to see submission of multiple characterization techniques to support the structure of the drug substance. Some of the standard techniques for small molecules include spectroscopic data such as NMR, UV-Vis, or IR. I would also like to note that for complex drug substances, we typically expect additional characterization. For example, for a peptide or oligonucleotide, we would expect characterization to confirm the sequence. As part of the drug substance characterization data, we expect submission of the raw spectral data itself. In addition to the raw data, we expect interpretation of this data as well. For example, assignment of peaks for an NMR or functional group assignments for an IR. During review of the IND itself, the FDA reviewers will then evaluate the totality of the characterization data and the provided interpretation, and will assess that information independently and provide any comments or developmental phase-appropriate advice moving forward. We acknowledge that identification of some peaks may be ambiguous early in development, as sponsors may be in the process of identifying impurities that are present at low levels. I want to briefly describe some of the physical-chemical characterization data that should be considered in the context of its criticality to the final drug product and drug product formulation. These types of attributes include physical attributes, including appearance and physical form of the drug substance. For example, is the drug substance a solid or an oil, and what color is it? We would want information on solubility in aqueous solutions, as well as any of the organic solvents that may be used as part of the manufacturing process. Information on particle size distribution may be limited at an initial IND submission, but the potential impact of this attribute for the drug product should be considered as additional clinical batches are manufactured. Information on polymorphic forms, as well as hygroscopicity, or the propensity of the drug substance to absorb water. Information on whether or not the drug substance is hygroscopic should be provided, and an assessment of the implications for the final drug product dosage form should also be considered. We generally encourage that sponsors monitor and characterize these attributes through development and assess the criticality of these physical and physical-chemical attributes to the final drug product, with an understanding that there may be changes to the drug product in early development. As part of the characterization section of the drug substance, there's typically an assessment of impurities. Generally, an impurity is any component of the drug substance that isn't the drug substance itself, and we typically want to minimize the presence of impurities in drug substances. Shown here is one relatively simple step of a drug substance manufacturing process to illustrate potential sources of impurities. Shown here is the reduction of a nitro group to an aniline using palladium catalyzed hydrogenation in the presence of hydrogen gas and ethanol as a solvent. From this one relatively simple step, you can see that there are multiple potential sources of impurities, including a heavy metal or elemental impurity, such as residual palladium. Residual ethanol could be a residual solvent that could carry forward to the drug substance. Any unreacted starting material would be considered a potential impurity, and in this case, the starting material has a structural alert, which is a functional group that may be of concern from a genotoxic perspective. In this case, the product of the reaction is also has a structural alert and could be a genotoxic concern. As a drug substance reviewer, we're essentially tasked with looking at the controls for the starting materials, intermediates, and final drug substance to assure that all of these potential impurities are adequately controlled and they don't pose any safety risk to patients. At early stages of development, we really want to know what control strategies are available for observed and potential impurities. And while we don't expect that all of these impurities are necessarily identified as part of the initial IND submission, we do want to understand the proposed strategy to further identify and control these impurities. And again, the emphasis in a phase one first-in-human study is really to address any differences in impurities and impurity profiles between the toxicology and clinical batches. There are a few overall categories for impurities and associated guidance documents that I'll briefly describe here. Organic impurities or process-related impurities are those that arise during the manufacturing process or during storage. The specific thresholds for reporting, identification, and non-clinical qualification of organic impurities are further outlined in ICH guidances Q3A for drug substances and Q3B for drug products. Genotoxic or mutagenic impurities and toxicological thresholds based on the duration of treatment are defined in ICH M7. I do want to point out that the scope of this guideline does not apply for advanced cancer indications or in cases where the drug substance itself is genotoxic. I also want to highlight the guidance document, Control of Nitrosamine Impurities in Human Drugs. Residual solvents are those solvents that may carry through from the manufacturing process to the drug substance, and the acceptable limits are defined in ICH Q3C. Elemental impurities may arise from reagents or catalysts as part of the manufacturing process or from the manufacturing equipment itself. The acceptable limits and appropriate tests are defined in USP chapters 232 and 233, as well as ICH chapter Q3D. And finally, microbial contaminants and limits are further defined in USP chapters 61 and 85. I would like to now just briefly describe some of the specific information that is needed for a drug product compared to a drug substance for early development. For the drug product, it is important that we have a description of the dosage form itself and any information to justify the safety of any novel technology or complex drug product formulation that may be used. Additionally, it's important to clarify any information that may be needed to support the administration of the drug product. For example, a drug product that has to be administered parenterally, the burden of data becomes greater as you have to address sterility and endotoxin concerns, and also consider any stability or compatibility issues associated with the administration devices or solutions for diluting the drug product itself. The quantitative composition of the drug product should be provided, which provides a list of the composition of the drug product per unit, including inactive ingredients or excipients. We ask that any information on the compendial status or quality of those excipients also be provided. If novel excipients are used, we would need additional information for the manufacturing and controls associated with that novel excipient to assure the quality of those excipients. And additional information may also be needed to assure safety of any animal-derived excipients, which may require further evaluation. The drug product manufacturing process should also be described with both a brief written description and a flow diagram. The flow diagram typically provides information on each unit operation and the in-process controls as part of the drug product manufacturing process. Additionally, information on the sterilization process should be provided in this section if it's applicable to your product. And finally, any degradation products or impurities that are specific to the final drug product dosage form itself. I'm going to now discuss information related to specification tests and stability in the context of both drug substances and drug product. The overall controls for release of the drug substance and drug product is essentially the controls part of chemistry, manufacturing, and controls, is essentially the specification, which is the predetermined set of criteria used to test and release batches for clinical use. The drug substance specification is unique compared to the drug product specification, but the overall format and presentation is pretty similar. The specification table essentially provides a list of all the test methods and proposed limits to assure that the clinical batches of drug substance and drug product meet the regulatory requirements and assure identity, strength, quality, and purity. In the initial IND, analytical test method can be very briefly described or referenced to a compendial test. The limits for an IND are typically flexible based on the available toxicology and clinical batch data, and are often further defined and narrowed as development proceeds and nears commercialization. Typically, we recommend that any critical attribute for the drug product be monitored as a test within the specification. For example, if the particle size of the drug substance is important to the drug product manufacturing, we would recommend that this attribute be monitored in the drug substance specification. As part of the IND submission, we expect to see either a tabular batch data or certificates of analysis for the proposed clinical batches tested according to the proposed specifications. And we also just want to point out that FDA is aware that specification tests and limits are likely to change as development proceeds. Shown here is a representative example of a drug substance specification table. Specifications include three main elements: the test attribute, the analytical method, and the acceptance criteria. We recommend that in the IND submission that you capture your rationale and thorough justification for the proposed specification, test, and proposed acceptance criteria. There's generally more flexibility with acceptance criteria earlier in development, with the understanding that there's very limited information and batch data available. What we're really looking for is that there's sufficient information to assure identity, purity, strength, and quality of the drug substance and drug product. I would like to briefly discuss the amount of data that's acceptable for an IND when it comes to stability. This is typically a very common question for the FDA. For an IND, preliminary data on representative material for both drug substance and drug product is typically acceptable for an initial first-in-human study. If stability information on the clinical batch is available, that information should be provided, and the stability section of an IND should clarify the tests that are run to monitor stability. If stability data is not available, we ask that sponsors provide a commitment to continue testing the clinical trial material through the duration of the proposed study. CMC issues in particular can often delay development if they are not addressed up front, and often these types of issues are likely to lead to major CMC safety issues and clinical holds of ongoing studies. The likely concerns include any question related to adulteration or purity of the materials used as part of the drug substance or drug product manufacturing process. These would be considered adulterated products. If the impurity profiles are not sufficiently characterized or the safety of the impurities is not justified by the supportive non-clinical studies. Any impurities that are of known or high toxicity that could pose potential risk to patients. Unreliable analytical methods that may undermine the confidence in the data provided in the IND. Insufficient batch data to demonstrate manufacturability of either the drug substance or the drug product. Any significant stability issues during the proposed storage conditions or during use of the drug substance or drug product. Lack of sterility assurance or endotoxin controls, for example, for parenteral drug products, would be significant safety concerns. Issues with the formulation, which may lead to formation of something like particulate matter on stability or during administration. And finally, issues with GMP at the facilities manufacturing the drugs. One way that these pitfalls could be avoided is by continued communication with the FDA through industry meetings and paying close attention to the advice given during those meetings. In particular, prior to an IND submission, every sponsor is granted at least one pre-IND meeting. As part of this meeting, the sponsor of that IND submits a briefing package that describes the background of the product and the proposed clinical development. Typically, specific questions are posed for each review discipline as part of the team. And for chemistry, manufacturing, and controls, those questions are typically focused on the general topic areas, including manufacturing process acceptability, whether the drug substance and drug product controls are acceptable, whether the stability data and overall stability protocol design is appropriate, whether the controls for impurities and adequacy of the toxicology batches and studies to support the clinical batches are appropriate, and any other overall potential gaps or hold issues that the CMC or product quality team may see. Beyond the pre-IND meetings, FDA also publishes guidance documents which provide FDA's current thinking on a topic. Guidance documents are typically recommendations and ensure that sufficient data will be submitted in a phase-appropriate manner. These guidance documents clarify the type, extent, and reporting of CMC information through development to assure integrity of the clinical studies to support marketing. I would like to end by providing references to some key guidance documents for submission of INDs through clinical development. And finally, I have two challenge questions for today's presentation. Challenge question number 1. The regulatory term for an active ingredient intended to furnish the pharmacological activity or other direct effect in the diagnosis, cure, mitigation, treatment, or prevention of disease, or to affect the structure or any function of the human body is A, drug product, B, impurity, C, drug substance, D, intermediate. The correct answer is C, drug substance. Challenge question number 2. The following are CMC safety concerns, except for which one? A, impurities of known or potentially high toxicity. B, insufficient batch data. C, lack of sterility assurance or endotoxin control. Or D, the manufacturing process route for the marketing application has not been finalized. The answer is D. All of the other options are potential CMC safety concerns. However, the manufacturing process for marketing is typically not finalized until closer to conducting the pivotal clinical studies. With that, I'm happy to answer any questions.
Thank you for the introduction. So we just heard about product quality information to support early drug development. And now we're going to switch gears and discuss pharmacology and toxicology information to support early drug development. My name is Matthew Thompson, and I'm a supervisory pharmacologist in the Division of Hematology, Oncology, Toxicology at FDA. The objectives of my talk are listed here. Given the short time I have today to discuss pharmacology and toxicology information, it is important for you all to know where to go to find additional information, and that is in large part going to be through FDA guidances. Now, the bulk of my talk is going to be about the types of non-clinical information you can expect to find in the investigator's brochure for a product. And I will continue to come back to this outline here of non-clinical information during the talk. So one of the most important links I can provide you today is at the top of this slide. And at this URL, you're going to be able to search for FDA guidances using keywords. And then below is a short list of guidance document topics with a letter designating the type of guidance and the number indicating the sequence. So, for example, here S refers to safety, and M, or in this case M3, refers to a multidisciplinary guidance. So you may find guidances helpful to both understanding what information you're seeing in an investigator's brochure, and also, if you're involved in development, what kinds of non-clinical studies are expected to support a clinical investigation. So without getting too detailed, I'll briefly say that in addition to the S for safety and M for multidisciplinary, there are also guidances which are Q for quality and E for efficacy. It's also worth pointing out that there's a chance you're going to be investigating a product in a healthy volunteer population. And so the M3 guidance is particularly relevant if you're going to be developing this type of product. Whereas the S9 guidance is very relevant to products used to treat patients with advanced cancer. And lastly, given the number of biologics we're seeing that coming into the agency, the S6 guidance is also worth a read. Now I'm going to shift gears and discuss the non-clinical information one could expect in the investigator's brochure. So Title 21 of the Code of Federal Regulations, Part 312, deals with investigational new drug applications. And 312.55 states the sponsor shall give each participating clinical investigator an investigator brochure. So what is in an investigator's brochure related to pharmacology and toxicology? Well, 312.23 discusses the IND content and format, and in A52 it states that there will be a summary of the pharmacological and toxicological effects of the drug in animals and, if known, in humans. Well, that's not providing a lot of detail, so we'll go into that in a little more depth during this talk. But before we start, one thing may be worth noting, and that's related to the terminology that I will be using. So at FDA, the word non-clinical is often used interchangeably with pharmacology and toxicology. So here is the outline of the non-clinical information in the IB that I'll be covering today. Pharmacology, safety pharmacology, and toxicology topics, including general toxicology, genetic toxicology, and other. First, we'll discuss pharmacology, and by this we generally mean information on the mechanism of action of a product and its activity, whether that be in vitro or in vivo. And we typically break that up into thinking about both primary, or the intended target and effects, or secondary, the unintended target and effects. So if we have a small molecule drug targeting a cell surface receptor, for example, I may anticipate certain toxicities based on this mechanism of action and how interfering with the receptor results in changes in signaling, leading to both the drug's therapeutic activity but also potential side effects. Primary pharmacology information may include binding assays, inhibition assays, data from cell lines, as well as the product's activity in an in vivo model of a disease. Secondary pharmacology will often consist of panels or screens involving many different enzymes, receptors, or ion channels to assess off-target effects of a product. And the type and amount of data varies based on a number of factors, including the product type. Is it a small molecule or a biologic? The indication, in other words, what's the patient population? Are we talking about a healthy volunteer study, or are we talking about a population with advanced cancer where life expectancy is shorter and available therapies are limited? And finally, the stage of development could influence the type of data available in the IB based on whether the pharmacology data is important to determining a first-in-human dose versus maybe later in development where understanding a toxicity that's been observed in a clinic may be informed by certain pharmacology data. The next area of non-clinical information is safety pharmacology. Safety pharmacology refers to studies designed to assess the potential of a product to affect physiological functions. And in practice, we are most concerned with assessing the effects of a product on vital functions. So therefore, the core safety pharmacology battery that you can expect to see information on is cardiovascular, central nervous system, and respiratory, since these are vital functions where acute effects could be life-threatening. Cardiovascular assessments may include in vitro assays to study the effect of a small molecule drug on a cardiac ion channel, and ECG data from a non-rodent species. Central nervous system and respiratory safety pharm studies are often conducted in rodents and will involve assessments of behavior, other neurologic parameters, and respiratory parameters such as respiratory rate and tidal volume. Moving back to our outline, we're going to be talking about toxicology and the first topic, general toxicology. General toxicology studies provide critical non-clinical information, allowing us to determine whether a clinical investigation is reasonably safe, whether the initial dose proposed in a clinical study, in other words, the first-in-human dose, is acceptable. General toxicology studies provide information on the potential dose-limiting toxicities that may be observed in human subjects and provide information on what to monitor for in the clinic. For example, a product may exhibit hepatotoxicity, so we may want to closely monitor liver enzymes and bilirubin. Or if it was cardiotoxic, troponins, or if we anticipated effects on the immune system based on the mechanism of action, cytokine levels. And very importantly, general toxicology studies in animals allow assessment of potential toxicities that we cannot readily identify in a clinical trial, namely pathology findings. So before I give you an example, I want to briefly describe the components of a general toxicology study. Again, this is brief. So a general toxicology study will provide information on the test article, the test species, the dose levels, the schedule of administration of the test article, the route of administration, the formulation, and other aspects of the design, such as the species, the age, the sex, etc. And the outcomes of the study will include mortality, clinical observations, body weights, food consumption, ophthalmology, ECG findings, clinical pathology, which includes hematology, clinical chemistry, and urinalysis. There's also gross pathology, organ weight data, and histopathology. When FDA reviews all of this information, we are looking at the totality of the non-clinical data, both the pharmacology and the toxicology, and we are assessing whether or not a proposed starting dose is acceptable and what safety signals need to be monitored for in the clinical study. So here's our first example. This is a biologic. It's an antibody drug conjugate being developed for the treatment of cancer. And in the animals, there was significant myelosuppression and severe hepatotoxicity that included both increases in liver enzymes and histopathology with finding of necrosis. So in a clinical trial, the labs would provide the information on the liver enzymes, but may not clue the investigator in as to the severity until it's too late, and the dose of the drug maybe has been escalated too high to a point where there's severe toxicity. Because we have the histopathology component from the animals, we can understand if the product causes a pattern of toxicity in a particular organ that includes that pathology, such as the necrosis, and tells us to be particularly careful in considering how the protocol is written. For example, knowing the potential for severe hepatotoxicity may affect the inclusion and/or exclusion criteria for the patient population in the trial. In considering the relevance of the non-clinical study to predicting toxicity in a clinical study, we consider two important things. The first is the design of the general toxicology study, with consideration of the route of administration and the schedule. And we're looking for that to be consistent with the clinical study. So for example, this is intravenous versus oral administration versus intramuscular, etc. And that this should match the planned clinical study, and the schedule should be consistent as well. So every day dosing, every other day, every week, etc. So it's important that the general toxicology study design provides the most valuable information. And consistent with this idea is the relevance of the non-clinical species. Typical non-clinical species used for evaluating products in general toxicology studies include two basic categories, rodents and non-rodents. So rat is the most common we see in the rodent group, followed by mouse. And typical non-rodent species are dog and monkey. So one thing sponsors will consider in the development of their non-clinical program and in the design of the general toxicology studies is what non-clinical species are relevant. And by relevant, I mean pharmacologically relevant. So a major consideration may be whether the target is present in that particular non-clinical species. And is the product going to interact with that target if it is present? We often run into this issue in dealing with biologics because they're so specific for targets, and a lot of the targets are not always found in rodents. So it's important that the selection of a non-clinical species is well justified and based on the science of both the target and the product. If the non-clinical species is pharmacologically relevant, then we are more likely to gain valuable information that can inform the clinical trial design, the investigator, and the patient about the risks associated with an investigational product. In this example, the small molecule drug, a microtubule inhibitor, is being developed to treat patients with advanced cancer. And this small molecule drug produced irreversible optic nerve degeneration in a rat study. As a result, clinical monitoring was increased, and information on this toxicity that was observed in rats was added to the IB and to the informed consent. And in this last example, a small molecule epigenetic targeting drug was found to produce malignancies in a 3-month repeat dose toxicology study in rats. The signal was also observed in the clinic, and appropriate steps were taken to address the finding. In this case, considering the patient population, 2 of the steps taken were the addition of the information to the IB to alert investigators and addition of the information to the informed consent to alert patients. The next topic in our outline is genetic toxicology. The area of genetic toxicology refers to the assessment of a product's potential to cause genetic damage. Since results from carcinogenicity studies can take a considerable amount of time to become available, genetic toxicology studies can be seen as a surrogate for carcinogenicity while clinical trials are ongoing, and this helps address concerns about risk to human subjects. A typical genotoxicity battery includes a test for gene mutation in bacteria, an in vitro chromosomal damage assessment in mammalian cells, and an in vivo genotoxicity evaluation. Timelines for genetic toxicology information to become available can vary. Typically, a gene mutation assay will support the determination of whether a single dose clinical study is acceptable. An additional in vitro chromosomal damage assay if proposing multiple doses in a clinical study, and a complete battery prior to phase 2. However, this can differ based on the indication and patient population. For example, if products are being developed in the oncology space, the full genotoxicity battery may be submitted with the marketing application. Now we're on the last topic, and though there are various toxicology studies that could fall under this other category, I'm going to briefly mention reproductive toxicology. An unfortunate example of drug-induced reproductive toxicity is thalidomide, which underscored the importance of using non-clinical models to assess potential for reproductive toxicity in humans. In considering the type and amount of non-clinical reproductive toxicology information in a regulatory submission, you can consider the patient population as well as the type of product. So is it a small molecule or a biologic? And you could even consider things such as the target or the mechanism of action. Reproductive toxicology studies covering fertility, embryo-fetal development, pre- and postnatal periods, you can generally look to the M3R2, S5R3, and S6R1 guidances. But based on the patient population, for oncology indications, you can also consult S9 and other FDA guidance. So to wrap up the talk, as you can see, we've really only kind of skimmed the surface of the pharmacology and toxicology information in an investigator's brochure. And in the non-clinical package that a sponsor will submit to the agency, there will be a lot more information. So if you'd like additional information on the types of non-clinical data that can be expected in a regulatory submission, you can consult the guidances and use that search function at that FDA website that I referenced at the beginning of the talk. Now, the pharmacology and toxicology, or the non-clinical data generated for an investigational product, is critical to taking an idea from the bench to the clinic, and from the clinic to an improved product that has activity and is safe. The mechanism of action of a product and its activity against a target, considering off-target effects, effects on vital functions, and the product's overall safety profile as assessed by general toxicology studies, as well as other types of toxicology studies, lays the groundwork for the product's development program, moving it from the bench to the clinic. Here's the first challenge question, and it's based on a guidance I've referred to several times during the talk. What FDA guidance is most relevant for non-clinical drug development in oncology? A, M3R2, B, S1B, C, M7, or D, S9? The answer is S9. And the second challenge question. The safety pharmacology core battery includes A, cardiovascular system, B, central nervous system, C, respiratory system, or D, all the above. The correct answer is D. All three of these systems form the core of the safety pharmacology battery. Thank you.
Good afternoon, everyone. Thank you for joining. Today, I'll be speaking to you about clinical pharmacology and how we fit into early drug development. Since the overall objective of my talk today is to explain a little bit about clinical pharmacology and discuss its critical role in early drug development, I'll be focusing on two main aspects. First, to understand the scientific underpinnings of clinical pharmacology as a discipline. And then, in the second part of my presentation, I'll be giving an in-depth overview of early clinical studies. There will be a focus on various aspects, such as study conduct, the information gathered as a result of these studies, and how to use that information in our drug labeling. And at the end, I'll also touch on a couple of developing areas in our field, including PBPK and model-informed drug development. Okay, so clinical pharmacology, what exactly is it? Like the old saying goes, if you ask 5 different clinical pharmacologists, you'll get 5 different answers. But in its simplest form, it is the study of the pharmacokinetics, or PK, and pharmacodynamics, or PD, of a drug in humans. PK is what the body does to the drug. It encompasses the processes of absorption, distribution, metabolism, and excretion for most drug products. PD, or pharmacodynamics, is what the drug does to the body. These two concepts are represented by these graphs on the right-hand side, where on the top graph you have a concentration of drug in blood or plasma, some type of biological matrix, over time. And in the bottom graph, you see an effect, some sort of biological measured effect over time. So what happens when we put it all together? We get a pretty powerful clinical pharmacology tool called a PK-PD relationship or exposure-response relationship, depending on the measured outcome. When we take a concentration over time profile and combine that with an effect over time profile, we get effect over concentration. So what we can really see is the effect of a drug over varying concentrations of the drug. And so in this way, we're able to predict some sort of quantified biological effect given any concentration of drug in the body. Given these tools at our disposal, how do clinical pharmacologists use them to contribute to the drug development process? Well, we like to say that we own the dose. And by that I mean we help determine the dosing regimen of a drug, how much to give, how often to give it. We even determine if a drug needs to be adjusted due to various intrinsic or extrinsic factors. In other words, are we giving the right drug at the right dose, at the right time, to the right patient? These are all very important questions that are asked of clinical pharmacologists and that we help to address through studies in early drug development. Now, I've mentioned ADME before, and assessing these processes gives us key information on the clinical pharmacology properties of a drug. The A in ADME stands for absorption, and it's simply the process of a drug reaching systemic circulation. It doesn't apply to all routes of administration. For an IV administered drug, there is no absorption since it's delivered straight into a vein and directly into systemic circulation. But absorption through an oral formulation, let's say, can be impacted by several factors in the body, such as eating food or pH-altering medications or other concomitant medications, alcohol, transporters, or drug metabolizing enzymes being altered in the gut. Important information that we typically communicate in the drug labeling, or USPI, in section 12.3 are the bioavailability of a drug, the time to reach maximum concentration, or Tmax, as well as the effect of food. The D in ADME stands for distribution. The distribution is the process of drug being distributed to different areas of the body, so it's just how it sounds. It is described by volume of distribution, which is a numerical descriptor that's given in the units of liters. It tells us the drug's propensity to either remain in blood or distribute to various organs throughout the body. So the higher the number, the more likely the drug is to distribute to various organs. Some factors that we need to consider for distribution are, does the drug bind to plasma proteins? Is the extent of protein binding either concentration or time dependent? These are important questions because only the free or unbound drug is pharmacologically active. Sometimes the measurement of unbound drug is recommended when interpreting data. In addition, is information on the distribution to cerebrospinal fluid relevant for this drug? Important information that we typically communicate in the drug labeling under section 12.3 include the percent protein bound of the drug, as well as the volume of distribution. Now here I've combined the M and the E. They stand for metabolism and excretion, and they fall under the general category of elimination. You may have heard that term before. Elimination is a broad category that includes metabolism and excretion. So metabolism in technical terms is the process by which lipophilic molecules are converted into more hydrophilic ones, so they can be excreted by the body. And excretion is the irreversible loss of drug from the body. So once it's excreted, it is not coming back. Factors to consider under this section is, how is a drug metabolized? How is it cleared from the body? This usually involves either hepatic metabolism or a combination of hepatic metabolism and renal excretion. And what fraction of the drug is being metabolized? And are there active or toxic metabolites that are being produced? Important information that we typically communicate in the labeling under section 12.3 include the half-life of a drug, the major route of elimination, and the percent excreted in either urine or feces or both. And here I have a few other important considerations for a drug. Is it a narrow therapeutic index product or drug? If it is, that means that very small or slight changes in drug exposure could significantly impact the drug's safety or efficacy, which then may require therapeutic drug monitoring in clinical trials and in clinical practice in order to minimize these toxicities and also to minimize the loss of efficacy. Also, is this drug a significant inhibitor or inducer of either cytochrome P450 enzymes or transporters? If so, this drug may need to be evaluated in further drug interaction studies. And now I'll move on to the next section of my talk, which is giving a brief description of early clinical studies that involve clinical pharmacology. First, I'd like to say a word or two about the timing of clinical pharmacology studies. We're often asked this question about when should I conduct them? Well, the answer is as early as possible. Right after data is collected from preclinical development and an initial IND is submitted that contains the first-in-human study in the U.S., we begin the start of clinical development. And during this process in phase 1, phase 2, and phase 3, the earlier you can get information from clinical pharmacology studies, the better, because in that way you can help inform the next study or the next phase that comes through, and then that helps maximize the chances of success for your drug. And early phase studies are usually designed to investigate the safety and tolerability, and sometimes to identify the maximally tolerated dose and the PK of the drug in humans. So with all of this information, this is critical for each step in the drug development process. So I mentioned that the start of an IND is the first-in-human study. So usually a first-in-human study entails a starting dose that's determined by some sort of preclinical tox study. And it includes a randomized placebo-controlled study in healthy volunteers. Sometimes patients are used in these studies, but that's fairly rare. And what we're looking to gather from a study like this is some initial safety and tolerability data, possibly the maximally tolerated dose, general PK characteristics of the drug, maybe it's highly variable, what is the PK linearity, and possibly some steady state parameters from the multiple ascending dose study. And preliminary exploration of drug elimination is also ideal. Next, we have the ADME study, which is recommended for nearly all drugs in development. Its objective is to understand the clearance mechanisms of the drug and any and all metabolites in humans. We just put out a new guidance on mass balance studies, and you can find that in the link here, or you can navigate to the FDA guidance website. It's sometimes referred to as mass balance studies, but a lot of folks also call it an ADME study. What it involves is you getting a radio-labeled drug molecule, administering it usually as a single dose to healthy volunteers, and using the intended route of administration, and then in the end, measuring concentrations of the parent and any and all metabolites to determine the relative amount of radioactivity in plasma and other biological matrices such as urine and feces. What we're hoping to learn from this study is to determine the overall pathways of metabolism and excretion for the investigational drug, finding out if there are any metabolites in circulation, and then determine if what the abundance of those metabolites are relative to either the parent drug or total drug-related exposure. Next up, we have bioavailability studies. It's important to conduct these in order to evaluate the rate, or Cmax and Tmax, and extent, or AUC, of absorption of a drug from a test formulation and then becomes available at the site of action. It's typically a crossover single-dose study in healthy subjects where the extent and rate of absorption of the parent drug and any major metabolites, if any, are measured. We can assess either relative bioavailability, which is testing one formulation versus another, or absolute bioavailability, which is the investigational formulation versus the IV formulation of itself. What we hope to learn is the amount of drug that reaches systemic circulation from a tested formulation. And we do have a guidance for industry that was just put out or updated last year on the FDA website. Moving on to my favorite kind of study, a food effect study. Since we all eat food every day, it is important to evaluate the impact of food on a drug's systemic exposure. So the overall objective is to evaluate the effect of food on the rate and extent of drug absorption from a given formulation, and this involves all oral formulations, of course. It involves a single-dose study in healthy volunteers using the highest therapeutic dose of a drug product. In the fed state should be an FDA high-fat, high-calorie meal, although other meals can also be studied. But the FDA recommended high-fat, high-calorie meal is contained in the new food effect guidance, which was just updated last year, and you can find that on the FDA guidance website as well. The PK assessments that come out of a food effects study are very similar to a bioavailability study. No food effect could be concluded if the 90% confidence interval of the fed to fasted ratios for Cmax and AUC fall within the 80 to 125% bounds. If an effect of food is found in a study like this, the clinical significance of it would be determined based on a drug's exposure response profile. What we would hope to learn from a food effects study is how to administer a drug in clinical trials, as well as how to label instructions on how to take the drug with respect to food, eventually in the drug labeling for patients and healthcare providers. So now I'll touch on hepatic impairment studies and just some brief points about it, such as when one should be performed. If a drug is intended to be chronically administered and is systemically available, if hepatic metabolism or excretion accounts for a substantial portion, which is greater than 20% of the absorbed drug, of the elimination of a parent or active metabolite, If it's a narrow therapeutic index drug, and this is irrespective of the proportion that's metabolized, or the metabolism route is largely unknown. If any of these criteria fit the investigational drug, then the FDA recommends that a hepatic impairment study be conducted. Additional details are listed in the hepatic impairment guidance, which is on the FDA guidance website. Speaking of organ impairment, next we have renal impairment studies. When should one of these be performed? Well, if a drug is likely to be used in renally impaired patients, and when we think that impaired renal function is likely to alter the PK of the drug or its active metabolites, because they are substantially eliminated by the renal route. If it's a therapeutic protein or peptide with a molecular weight less than 69 kilodalton, or drugs that are likely to be used in patients with end-stage renal disease undergoing dialysis, they should be evaluated both while the patient is on dialysis and off dialysis in order to determine if the contribution of the dialytic method to the elimination of the drug and its potentially active metabolites. These details, as well as others, are set forth in the new updated renal impairment guidance that was just put out 2 years ago, or 3 years ago, on the FDA guidance website. So one very important aspect of clinical drug development is the assessment for the potential of a drug interaction with the investigational drug. Now, the investigational drug can be either the drug being impacted, or it can be the drug that's altering the PK of other drugs. So first, we start with in vitro tests or screening to determine if the investigational drug is a substrate for or an inhibitor or inducer of common drug metabolizing enzymes and transporters. These are set forth in the guidances for industry on the FDA guidance website. We've divided it into 2 guidances, one for in vitro and one for in vivo, and these were just updated in 2020. And some of the more common enzymes or drug metabolizing enzymes are CYP3A4, 2C9, PGP is a very common transporter that we recommend screening for as well. If any of these initial screening tests are positive, we recommend conducting an in vivo drug interaction study to confirm the involvement of the drug, whether the drug is a modifier or it's the substrate. Then, based on the results of these drug interaction studies, it can have some wide implications for drug labeling. These implications can range from something informative such as drug X is not a substrate for CYP3A-mediated metabolism, all the way to a contraindication for the use of this drug with other drugs. In order for drug interaction studies to be informative, they need to be designed very carefully. There are some key points that need to be considered when conducting a drug interaction study. Some of these would be factors that need to be taken into account in order to maximize the possibility of detecting an interaction. So keep in mind that we're trying to put forward a worst case scenario, if you will, so that we can find out if there is a true potential for a drug interaction. So we need to select a proper dose for the inhibitor or the inducer. We need to think about the route of administration, the timing of the co-administration of the 2 drugs, as well as the number of doses to be given of each of the 2 drugs. The degree of the effect, either inhibition or induction of a drug metabolizing enzyme, is typically classified by the change in the substrate's AUC. So it could be a weak inhibitor, a moderate, or strong inhibitor. Here, the exposure response profile, again, is important for the drug in order to assess the clinical relevance of any change in AUC caused by either an inhibitor or an inducer of the enzyme of interest. So next, I'll just touch very, very briefly on physiologically based pharmacokinetics, or you may have heard PBPK. PBPK modeling is grounded in what we know to be true of human physiology, and there are a lot of inputs that go into these models. In theory, this approach allows us to make predictions based on our knowledge from previous trials and previous in vitro investigations, and also sometimes preclinical studies. So what exactly is it? PBPK is a mechanistic modeling approach which utilizes all of these inputs, preclinical, in vitro, in vivo data, to help us predict the behavior of drugs in humans. And what we typically apply this to is to explore the effects of various intrinsic and extrinsic factors on human PK. Some of these factors can include, age or ethnicity, the underlying disease status, or even sometimes drug interactions. So another method we use in order to make some predictions of drugs without directly and empirically testing it is model-informed drug development. And I guess in technical terms, PBPK can actually fit under this larger umbrella of MIDD, because MIDD is an approach that involves developing and applying exposure-based biological and statistical models derived from preclinical and clinical data sources to inform drug development or regulatory decision-making. So it's a pretty broad umbrella. And also having well-timed and well-designed dose-ranging studies or dose-finding studies are important for avoiding dose selection pitfalls later in development. And MIDD can help with these dose selection type of issues. A few years ago, the FDA initiated an MIDD paired meeting program that allows drug sponsors and drug developers to meet with the review team, which is, in the case of MIDD, led by clinical pharmacology. Some of the focal points or issues that have been discussed in the past have been dose selection or estimation, clinical trial simulation that's based on drug trial disease models. These may be used to inform the duration of a trial or even selecting appropriate response measures, predicting outcomes, etc. We've also had discussions about predictive or mechanistic safety evaluations. In these cases, we've used systems pharmacology and other mechanistic models for predicting safety, and also identifying critical biomarkers of interest. So we've had a lot of successful cases of MIDD already. And last but not least, I'd like to acknowledge Kelly Reynolds for the use of some of her slides, and Leonard Sachs for the invitation to do this presentation, as always. Thank you.
Well, thank you very much to our panelists for excellent introductions. I really cannot overemphasize how fundamental these sort of preclinical and non-clinical issues are in the development of drugs. I think we all remember that yesterday in her introduction, Kim spoke about diethylene glycol and the catastrophe that resulted when it was used as a solvent for sulfanilamide. And from Matthew, we heard how modern, at least from Pinky, we heard how modern CMC is targeted at addressing all these potential problems. And we also heard from Kim yesterday about the thalidomide catastrophe. And here we heard from Matthew about toxicity studies and reproductive toxicity and so on. So I think these are all obviously very fundamental to ensuring that clinical trials are safe. So I'm going to turn to the panelists, ask them to come back on, and we're going to start with questions. We have a whole bunch of questions from the audience here. So here's for Dr. Patel. Pinky, maybe we can start with this one. Let's see. What are the GMP expectations for a phase 1 study at the time of an IND submission? Any thoughts there?
Thanks for that question. So for phase 1 studies, there is actually a guidance in that focuses on GMP requirements. I think it's titled CGMP for Phase 1 investigational drugs. And what I would say is that Phase 1, we don't have to explicitly follow the CGMP regulations that are outlined, and those are outlined in 21 CFR 210 and 211. However, the guidance does clarify the 3 major areas for focus for manufacturing of Phase 1 investigational drugs, which kind of outlines that you need to have well-defined and written procedures and adequately controlled equipment and manufacturing environments, and that you need to have recorded, accurately and consistently recorded data from the manufacturing as well. However, as you move into Phase 2 and 3, those studies do need to be conducted and are subject to the parts of the CGMP regulations in 210 and 211. So hopefully that kind of answers the question about the GMP requirements throughout development. Thank you.
Thanks, Pinky. I think that certainly covers the Phase 1 studies. I'm going to keep you on the spot a little bit because a lot of people had questions, and I think some of them may be fairly direct and easy to answer. Here's another one for you, which is, what is the level of purity required for pharmacological products? Is that something you can address?
Thanks, Leonard. Thanks for that question. Yes, this is a good question because there's really no regulatory requirement or that actually outlines explicitly the purity required for a drug substance or a drug product. And one of the reasons why is that, as you can imagine, in my presentation, I describe some of the molecular variability that we see for drug substances. So oftentimes those purity considerations may vary depending on the source of the drug substance and how that drug substance is isolated, whether it's manufactured using chemical synthesis or whether it's isolated from natural sources. But I do want to point out that the emphasis for a Phase 1 study is really the difference in purity and the impurity profiles between the toxicology batches that are used in non-clinical studies and in the toxicology studies, and then those batches that are used for proposed Phase 1 clinical studies that are provided in the IND. So that's what our focus is when we're looking at the differences in quality. But I guess, to give a little baseline for something like a small molecule, typically we would expect something that is higher than 95% purity if it's chemically synthesized in with a short chemical synthesis. However, something that's larger and inherently heterogeneous, may be isolated from a natural source, then those purity requirements may be lower than a small molecule. So hopefully that answers the question. Thank you.
Yeah, I think that's a very sort of comprehensive way of dealing with the purity. Just as an extension of that, or at least a little beyond purity, the next question is what aspects of excipients are important for an IND submission? So any thoughts about excipients there in an IND?
Yeah, this is a really great question as well. So excipients are essentially the other components of a drug product that is not the drug substance itself, right? These are the components of a drug product that are necessary to formulate the drug substance for delivery to the patient. So for an initial submission, we would typically want to know what the quality of that excipient is. Many excipients actually are compendial in nature or in quality, and we ask that that reference to the appropriate USP or National Formulary Monograph be provided in the submission if it's a compendial excipient. If the excipient is novel, meaning that perhaps it hasn't been tested in humans previously or there's no approved products with those excipients, then in those cases we would need additional chemistry information about how that novel excipient is manufactured, what controls are in place to assure its identity, purity, and quality. So that's, we would need additional information for those novel excipients. I do want to point out that the agency has this inactive ingredients database, and this database provides the levels of excipients in approved products. So typically we ask that sponsors look at that database and kind of provide a justification for why the levels of the excipients that they may be dosing patients, why those levels would be justified based on those inactive ingredient database levels. In addition, there may also be additional non-clinical or toxicology data that's submitted for those excipients as well to justify their proposed dosing and levels. Thank you.
Thanks very much. I think as clinicians, many of us probably give not enough thought to the excipients, and we can focus on the drug substance. So I think these are all very critical considerations. I guess let's turn to Matthew. Matthew, there's a question for you here, which I think is an interesting one regarding the design of toxicology studies. The questioner asks, what is an appropriate duration for the dosing in non-clinical toxicology studies to support clinical drug development? Any thoughts about that?
Yeah, so the design of the non-clinical studies should be appropriately chosen in consideration of the clinical dosing. So if you're thinking about a certain schedule in the clinical setting, you can basically match or have the that dosing regimen reflected in the design of the non-clinical studies. And you can also kind of leverage some of the information you learn in your initial preclinical or non-clinical studies, for example, on PK, to try and understand half-life, to try and understand, how your product behaves in an in vivo system, to understand what you think, like, are you hitting a target level for the drug, and then take that information, take that plus maybe the PK, take that, so basically a PK/PD, and then understand, there might be other risk-benefit considerations for the population, and then you consider the design of the non-clinical study, and that it's going to hopefully reflect what you're going to plan to do in the clinical study. And there's tables in the S9 guidance and the M3R2. So the S9 for oncology and the M3R2 for the non-oncology indications. I think they're both table 1s. And so those tables kind of could give you an idea of how to make sure that your clinical dosing is reflective of your non-clinical, and vice versa.
Thanks very much. And would there be circumstances where you would buffer it by, for example, prolonged exposure? Just a thought.
Yeah, so we would generally hope to see that the non-clinical studies would basically provide the same, if you will, dosing intensity as you'll see in the clinic. So if you're thinking you're going to dose, let's say you start and you're not dosing it as intensely, it's more spread out, but you're anticipating it becoming more intense, then you'd probably want to. That's where you kind of have to. It's tricky. You have to kind of think about how you would design your non-clinical studies to provide that kind of coverage, so to speak, for the potential development of the clinical product. Yeah.
Right.
And actually, I should add one more thing. Would be, we do expect different durations of studies. So if you're going to dose, it's a little different in oncology because a lot of times therapeutics are dosed until disease progression, for example. And so we would generally say like a 4-week repeat dose tox study would provide coverage for the initial clinical development, and then we'd want 3-month studies to support a registrational trial. And that those 2 dosing durations for repeat dose tox studies would cover development for a typical oncology product under S9. But if you were to talk to somebody outside of oncology looking at an M3R2 guidance, they would want longer duration studies to provide coverage for longer duration studies in humans.
Yeah, I think that's a very comprehensive answer. Obviously, it's going to depend. I mean, there are drugs which are used at a single dose, and there are drugs that are used for basically the rest of a person's life. So yeah, I think these things have to be tailored. While I've got you on, Matthew, there are several questions. Let's try this one. This person asks that if toxicity is observed for an organ at a dose several folds higher than the no adverse event level, and the toxicity is not seen at the no adverse event level, does this observation get communicated to the clinical reviewers? So if there's toxicities that are seen at high levels that are not going to be used, do we tell clinical reviewers about this so they can look out for toxicity?
Yeah, so that's a good question. I would say my first comment would be we reviewed products as a team. So we have clinical and non-clinical and ClinPharm and CMC and stats, and we all get together and we have actually group meetings or safety meetings, and we share all the information that we've learned when doing the review. And so if we were to see a toxicity in a particular organ, let's say liver, at a higher dose than the NOAEL, and in oncology we use different benchmarks for dosing than NOAEL usually. But if let's say we see a clean tox profile at a certain dose, then at a higher dose we see liver tox. So we would absolutely communicate that to clinical, and we'd want to make sure that, as dose escalation, you can always, animal studies do not necessarily translate always to the human setting. So we would certainly communicate that. And in the clinical protocol, for example, if you communicated that to your clinical team, they would just make sure that there would be labs to measure for, for example, liver enzymes, and that the protocol is going to adequately monitor that. So really, it's any signal that we see in a non-clinical study that we think could potentially be observed in the clinical setting, especially if it's severe in nature. And that's true. Even if you see a clean dose level, a low dose, you don't see this toxicity. If you see it at a high dose and it's severe, for example, then you would certainly communicate that to the clinical team, and there would be an effort to make sure the monitoring and the clinical study is adequate.
Thanks very much. Yeah, that's definitely been my experience back as a reviewer, where I think we're all very sensitive to any signal that's found in the animal studies. Good. So let's give Shirley a crack at this one. The question is, when during drug development would you recommend a food effect study, and why? So when food effects, and why would you request it then?
Right. Thank you, Leonard, for the question. So a food effect study is important as early as possible in drug development. And the reason is because as you move forward into phase 2, phase 3, you want to know how to instruct study participants on how to take the drug, whether it should be given in the presence of food or without food. And then ultimately that will translate into the drug's labeling if and when the drug gets approved. So we typically, tell drug developers that the earlier we get this type of information, the better it is for them. Thank you.
Great. Now, another question for you, Shirley, which certainly looks interesting and thought-provoking. Are there any notable differences in the IND process for botanical drugs that should be considered? Any thoughts about botanicals?
Yeah, right. So I think this question may have been targeted to Dr. Patel.
I beg your pardon. Yeah, I think it is, yeah. Pinky, would you mind answering that one? I'll repeat it for you. Are there any notable differences in the IND process for botanical drugs?
Yeah, thanks for that question. So I would actually point to a guidance that we have there for industry. It's related. It's the title of that guidance is Botanical Drug Development Guidance for Industry, and this guidance actually kind of outlines some of the requirements for INDs for botanical drug development throughout the development process. There are some notable differences in considerations for botanical drugs, and this guidance kind of outlines some of that. But I would say one of the major parts of the botanical quality considerations is that the whole botanical drug substance that's isolated from that manufacturing process is considered an active component. So as part of that, it's important to try to understand all the different molecules or all the potential ingredients that are part of that botanical mixture. So from a CMC perspective, these are inherently challenging drugs, and they're heterogeneous as well. One of the key parts of a botanical that is required for an IND is information about the actual raw materials and the controls for that raw materials that are the inputs for the ultimate drug substances manufactured. And there is a USP chapter 563 that defines some of the requirements for the identification of those sorts of products from botanical origin. And so usually what we will do is make sure that the botanical raw materials meet those USP requirements. Beyond that, typically for an initial IND, we do want to have an understanding similar to what was presented as far as the manufacturing process and the controls in place. And we would provide a little bit more information after that original submission related to the recommendations for assessing mass balance of the botanical drug substance as well. And so I think the overall outline of the requirements are really in that guidance. So I'm gonna point the person to this guidance, but I do want to also point out that one of the important parts of botanicals is also there's typically a prior history of use. So we kind of want to see what information is known as far as any information about use, prior history of use of that botanical in humans to support the original IND submission. Hopefully that provides some information on botanical drug development. Thank you.
Thanks very much. I think it's obviously a very complex issue because these are combinations of many different moieties. Shirley, getting back to you, here's a reasonably straightforward question. How many subjects are usually present in a first-in-human study? Any thoughts about how many you need in a first-in-human study?
Yes, thanks, Leonard. I would say the vast majority of first-in-human studies involve somewhere between 6 and 12 healthy subjects per cohort. Now, the number of cohorts can vary greatly depending on whatever scientific questions that the drug developer is interested in answering. So in my personal experience, I've seen first-in-human studies include a dozen or so cohorts, several single ascending doses being tested in different cohorts, and then they kind of move on sequentially to multiple ascending dose cohorts. And sometimes they even wrap in an initial food effect evaluation. So the total number of subjects in that one sort of large first-in-human studies can be quite a large number. But if you're looking to just test one dose and one cohort, typically it's in the single digits, between 6 and 10, sometimes 12.
Thanks. And I do have another question for you here. Let's see. This is about when to do your ADME studies. It says the draft mass balance study guidance indicates that studies should be conducted at the latest before initiating late phase trials. However, the person who's asking the question says he's heard that ADME studies should be conducted before beginning large scale trials. So I guess the question being asked is, what is considered a large-scale trial? Would a phase 2B with exposure of over 100 subjects be considered large-scale? Would you need ADME studies before those? Any thoughts about that?
Great question. Thank you. In general, the timing of an ADME study is critical to help de-risk drug development and overall across all phases. So the more initial knowledge that we have on the way and the extent to which a drug is absorbed, how it's metabolized or excreted, the more information we have on those characteristics, the better. And the reason is, as you start moving into, quote, large scale, and I would, probably consider 100 subjects to be somewhere in the middle of the road. I wouldn't necessarily call that a very large scale study, but it also depends on the condition or disease that's being treated. For some conditions, 100 might be considered quite large, whereas for maybe a cardiovascular trial, 100 seems quite small. But if the drug is metabolized to some extent, you want to know how is it being metabolized and how much. In that way, you might be able to help mitigate some drug interaction concerns when they pop up in the inclusion and exclusion criteria for a phase 2 study or phase 3 study. That's very important information to know, so you're not confounding the results of that study. Thank you.
Just a quick question for my own clarification. So why would you want to keep it down to a limited number? Is the concern that without ADME studies, you wouldn't know enough safety to expose a lot of people? Or are there other reasons why you would not want to go into a big study before doing the ADME?
Right. Yeah, I meant to point that out as well, is that, safety is a big concern. So without a lot of, detailed knowledge about the ADME, yes, I think some, drug developers might approach these studies, very cautiously, and so, include a very small number of subjects in the beginning before much is known. So that's why you see these first-in-human studies as a very small initial cohort sample sizes. So those numbers are quite small in the beginning until you start accruing data. And starting to find out what is a tolerable and safe, somewhat safe dose. Yes, the total number of subjects you want to expose should be on the lower side for safety purposes.
Great, thanks. I'm going to turn back to Matthew. I've got a tricky question here. Let's see if you can deal with this one. The question is, are only the animal studies conducted in a GLP facility acceptable for general toxicology data in an IND? In other words, can you use other data that's not conducted in a GLP facility? Any thoughts there?
Yeah, so let's see. So for animal study results, I could just touch on, let's just think about what kind of study results we get in. So you'd see in vivo pharmacology, not expected to be GLP, just to comment on that. For ADME studies, generally, no, they're not going to be GLP. Single dose, and then under general tox, you would typically think of things like single dose studies. So those, and also dose range finding studies, those will typically not be GLP. So a company will want to start initiate a pivotal repeat dose toxicology study. Let's say it's a 4-week study. They want to be kind of careful because they're going to put a lot of money into that GLP study. So they're going to probably do a study where they're going to do a dose range finding study to try and figure out where that MTD is in the animals, in their non-clinical test species, so that they can not go into that pivotal study and have a result in a lot of morbidity and mortality. So there's certainly, you see a lot of those studies submitted. They're usually non-GLP, and so really the only studies we typically see or that we typically expect in a submission to initiate an IND would be the pivotal, like for example in oncology, it would be the 4-week repeat dose toxicology studies would be the GLP studies, but that being said, it does depend on the product and the nature of the product. There'd be considerations about relevance of non-clinical species. There'd also be consideration of the dosing regimen. So if it's a single dose, one time only, then obviously the duration of the study would be different than if it was something that was going to be continuously dosed. So yeah, I would say it's pretty fair to say that the pivotal general tox studies are going to be the ones that are conducted in a GLP facility.
Great. I think that's a very sensible answer. While you're on, Here's another interesting question, lots of interest in toxicology and animals. Could you please give examples of when certain types of non-rodent data are warranted? Any thoughts about non-rodent data?
Yeah, non-rodent. So you just break up, I mean, outside of, so number one, actually, one thing to consider is you really should, the best advice would be to contact the agency if you have questions, because if you talk to somebody in oncology, they're going to give you a different answer than somebody outside of oncology. And that's a reasonable thing, because the risk benefit for the patient population you're going to be studying this in is going to be different. And the oncology, typical oncology patient, there's a different risk benefit profile there. So we would, so first of all, think about that. Think about where you're going to submit this to. Contact the folks you're going to submit. You're thinking about submitting the IND to. What's your product? Is it a biologic or is it a small molecule? Because that's going to kind of partly answer this question. So if it's a small molecule in oncology, for example, we typically expect 2 species. It would not be typical to see a non-rodent species. It wouldn't be typical to see non-clinical test species be considered pharmacologically not relevant for toxicity testing. So for small molecules, we'd expect a rodent and a non-rodent to characterize on-target, off-target toxicity. And then for certain cases where types of non-clinical data are warranted, let's see, I would say, so for small molecules, again, it's going to be rodent, non-rodent. And then for biologics, that's where you kind of really start to kind of ask the questions of, is there binding? Is there activity? Is there expression of the target? And so in those cases, that's definitely case by case. And that's something you have appreciated about the agency, is we definitely consider things in a case by case way. So, and if you read guidance documents and stuff, you kind of get that sense that there's going to be certainly some, especially in oncology, regulatory flexibility and case by case. So, for a biologic, you might not have a target expressed in a rodent, or there might not be binding in a rodent, and therefore you wouldn't see that a study done in a rodent. You'd only see studies done in, for example, a monkey. That would be the most classic case for a biologic. So I think that kind of answers it. So it really depends on what division you're going to, probably the indication. It depends on the type of product, and then it depends on the real scientific basis of it. So what's the justification for using a non-rodent versus a rodent? Is there again, is the pharmacology there? Is there a pharmacologically relevant reason for why you would test it in a non-rodent versus a rodent? And the expectation, like I said, generally is for small molecules, 2 species. For a biologic, 2 species. But a lot of times we're seeing biologics that just there is not a relevant test system for the rodent, and so you just see it in a non-rodent.
Yeah, thanks very much. I'm assuming that the whole purpose of non-rodent species is to try and get as close as you can to the human model, and
yes.
I presume that there are some instances where really it's very difficult to do anything short of a primate. So maybe those are other considerations.
Yeah, and actually we have seen even recently now products approved that they have no relevant species for toxicity testing, and they are approved based pretty much on pharmacology data and have no toxicology data in the submission. So it's certainly where you want to have the non-clinical data should be justified why you should think about why you're doing it. And if it's you're doing a study and it does not have relevance to the clinical setting at all because it doesn't bind or the target's not expressed, then you should. I mean, that would be a case where contact the agency and see what the agency thinks. See if there's agreement, if there's alignment there, because you do want the non-clinical data to inform the clinical trial. If you don't, then there's not a lot of value in that. So.
Yeah, excellent answer. I know we've got till the top of the hour, so I'm going to squeeze in 1 or 2 more questions. Shirley, one for you. How can computational modeling and simulation be used to optimize drug pharmacodynamics?
Yes, thank you, Leonard. Great question. So when you have enough data to develop a relationship between the time course of drug concentrations and versus a PD effect or some sort of clinical response, you can build an exposure response relationship. And using this modeling, you can predict favorable or non-favorable PD responses based on a given drug concentration. So what you might know about this drug's Cmax or C average or area under the curve, AUC, et cetera, you can then kind of relate that back to a dose. And so once you have this, dose and exposure response relationship, you could very easily identify an optimal range of exposures and then dose for your next trial. So given a known favorable PD effect, you then, correlate that to a range of exposures that you want to target, and then you'll know what dose you should be giving to patients, or it may be a range of doses that you should be testing next, or maybe identifying coming out of your phase 3 trial for approval.
Right. And I presume these models can also be used for products with nonlinear pharmacokinetics?
Oh, yes, absolutely. It does get a little trickier as the, PK gets a little, irregular, I suppose. But yes, there are definitely models that can address all of the, irregularities with a drug's PK characteristics. We've encountered many of these, and in most cases, we've been able to derive a fairly robust exposure response or PK/PD model. Sometimes we're not able to, and it's usually due to the paucity of data. We need a good amount of PK data in order to inform a robust model.
Great. Thanks for a comprehensive answer there. One last question for Pinky here. The question is it necessary to provide FDA with regular updates on the drug product stability profile during the course of the trial? For example, data obtained at each testing time point and after dosing the last participant. So do we need multiple tests during the course of a trial of drug stability?
Thanks for that question, Leonard. Yes, so short answer is yes. We do need, FDA does expect stability data updates for the clinical trial material or clinical study material that's used beyond phase 1 studies. So in that original IND submission, we expect the preliminary data that is available either for the clinical batches or even supportive data for maybe the toxicology batches, for example. But beyond that phase 1 study, we do want to see the clinical stability data for the batches that are being dosed to patients. Right, we want to assure that those batches that patients are receiving continue to be within the quality specifications that are set for that product, and that quality maintains throughout the period of time that patients are being dosed. And so that the stability requirements during clinical studies are outlined in the CMC guidances for phase 2 and 3 studies as well. So beyond that phase 1, kind of having those updates and typically those updates are submitted as IND amendments or as annual reports to the IND. Thank you.
Great, thanks very much. Well, I think that brings us to the top of the hour, so I want to end with a vote of thanks for the great panel, great discussion, excellent questions from the audience. We're going to take a 15-minute break, and then we're bringing you back for another hot topic, which is clinical trial quality, investigator responsibilities, and site inspections. And afterwards, we will deal with international trials. So thanks, everybody, and we'll see you at 15 minutes past the hour.
Welcome back from the break. We're on the home stretch now, right into our first presentation on investigator responsibilities, regulation and clinical trials, and clinical trial quality by Ann Meeker O'Connor, Director, Office of Clinical Policy, Office of Clinical Policy and Programs in the Office of the Commissioner. Next, we'll learn what to expect on clinical investigator site inspections. Dr. Stephanie Kokia, Good Clinical Practice Assessment Branch, Team 1, Division of Clinical Compliance, Evaluation, Office of Scientific Investigations, Office of Compliance. And our final presentation before the Q&A session is on international clinical studies by Dr. Kasa Yalu, Branch Chief, Division of Clinical Compliance Evaluation, OSI, in Cedar. Please join me to welcome our first presenter, Director Meeker O'Connell.
Good afternoon, everyone. My name is Ann Meeker O'Connell, and I serve as the Director of the Office of Clinical Policy here in the Commissioner's Office at FDA. So I'm going to be talking to you today about integrating quality into clinical trials, which is quite possibly one of my favorite things to talk and to think about. So in the clinical research enterprise, we often think of quality as an activity that exists separate from the design or the conduct of a clinical trial, perhaps as a separate function or department. In reality, quality is everyone's role that's involved in a trial, and quality is delivered through the decisions made and the actions taken along the way as the trial is in progress. And those of you who are serving as a clinical investigator have a really important role in delivering quality, as it is your work, day in and day out, whether that's in engaging participants, in carrying out protocol requirements, or collecting trial data. Those really form the bedrock of a trial that yields reliable data and reliable conclusions. So today, we're going to talk about the regulatory perspective on clinical trial quality. We'll identify the federal regulations that govern clinical research and touch on some key clinical investigator obligations. And we'll close with talking about some ways, some methods that you can use to enhance compliance with the federal regulations, with study protocol requirements, and ultimately deliver quality clinical trials. Oddly enough, you won't see the word quality defined in our IND regulations, although part of the reason FDA reviews protocols for IND studies prospectively is to assure that the quality of the scientific evaluation of drugs is adequate to permit an evaluation of the drug's safety and effectiveness. To understand quality in the context of how trials are conducted, we need to look to guidance. In this case, ICH E8 Revision 1, which talks about general principles for clinical trials. So ICH E8 defines quality as fitness for purpose. And what that means is that if the trial is intended to support product approval, the trial design, its conduct, and the data that are generated are fit for regulatory decision-making. This concept of quality as fitness for purpose extends from design into conduct in the latest revision of ICH E6, the Good Clinical Practice Guidelines. ICH E6 Revision 3, or R3, emphasizes proportionality and risk-based approaches, really looking at both quality in terms of keeping the emphasis and focus on the reliability of trial results and participants' safety. The intent here is to make sure we're minimizing unnecessary burden on participants, on sponsors and investigators through really keeping a keen focus on what matters most to quality and what matters most to participant safety.
with at the end of the day a goal to yield efficient and reliable trials. In thinking about minimizing burden and focusing resources on what matters, a good example would be the draft E6R3 makes clear that there's no blanket study training requirement, and that training should correspond to the role that an individual is expected to take in the trial. At FDA, we refer to our regulations that govern human subject protection and the conduct of clinical trials as our good clinical practice regulations. These govern the appropriate design, conduct, analysis, and reporting of trials. So if I had to distill the entirety of GCP from our regulations, from the ICH guidelines down to three things, it would be that a trial is well designed. That is, it's of appropriate scientific quality, and the design itself is ethical. Second, that those conducting the trial adhere to ethical requirements for the protection of participants' rights, safety, and welfare, including obtaining institutional review board review and approval, and obtaining informed consent from participants. And lastly, that the trial generates reliable data that can be used to drive decision-making, whether by a regulator, a clinician, or another party. So those three things, to me, again, are good clinical practice. If you delve into our regulations and guidance, it's clear that multiple parties play important roles in trial quality. This includes sponsors who design trials, who oversee them, who analyze and report results. It includes contract research organizations to whom sponsors have transferred their responsibilities, as well as IRBs who provide initial and continuing ethical review and approval of research. And of course, those of you who are clinical investigators who translate a trial from the pages of a protocol into practice in the clinic are critical to quality. So, under our regulations, who is an investigator? The clinical investigator on a trial is the individual who actually conducts a clinical investigation, that is, under whose immediate direction the drug is dispensed to a participant. If there's a team, the investigator is the responsible leader of that team. Clinical investigators are really at the center of it all, and may engage with many different parties in the clinical trial environment. So if you're a clinical investigator, you serve as the key connection point between the participant and the trial. And to be able to start recruiting for a trial, you need to engage with the IRB for their approval and to communicate with them throughout the trial at various points. The sponsor has an obligation to select qualified investigators and to provide them with the information needed to conduct the study appropriately. And they also have an obligation to oversee trial conduct. So that means as an investigator, you'll engage with sponsor and or CRO staff from initial study startup through closeout of the trial at your site. FDA has a really important role to play in overseeing trials and verifying that data submitted to us are reliable and that participants are protected, which we do through both reviews of protocols and also on-site inspections. If your trial is federally funded, the Office for Human Research Protections, as a sister agency, may also be engaged in oversight of the human subject protection aspects of your clinical trial. So again, it is a very broad range of people that it takes to be able to carry out research. That may sound like a lot, and you may be sitting thinking to yourself, can I have a co-investigator? The answer is yes and no. Yes for your needs, but no for regulatory purposes. Each quote-unquote co-investigator would be fully responsible for fulfilling all obligations of an investigator and would need to sign a separate Form FDA 1572. Another question we frequently receive is whether the investigator has to be a physician, a medical doctor. The answer is no. The investigator needs to be qualified by training and experience as an appropriate expert to investigate the drug, but is not required to be a physician. A physician can be a sub-investigator if the clinical investigator is not medically trained, so that there's somebody there to perform study functions that require the appropriate level of medical expertise. So I imagine that out there are some of those in the audience who may aspire to both design and initiate a trial, but also to conduct it. That is, to play the role of both study sponsor and clinical investigator. Our regulations define this as a sponsor investigator, an individual who both initiates and conducts an investigation, and under whose immediate direction the investigational drug is administered or dispensed. So in this case, you would need to know and adhere to regulatory requirements that apply to clinical investigators, which we'll walk through in the discussion today, as well as those that apply to sponsors. So sponsor investigators may also have regulatory obligations outside of those defined in FDA's regulations for clinical trial registration and results information submission to the clinicaltrials.gov databank. This requirement is covered by 42 CFR Part 11. So this is a different Part 11 than FDA's, which is focused on electronic records and systems. This Part 11 is a final rule issued by NIH and HHS that implements the provisions of Section 402J of the Public Health Service Act as amended by Title VIII of the Food and Drug Administration Amendments Act of 2007. So it clarifies and expands on the requirements for the submission of clinical trial registration and results information for certain applicable clinical trials. So this final rule was effective in January of 2017 when modifications to the clinicaltrials.gov databank were implemented, allowing responsible parties for certain applicable clinical trials to comply with the final rule. So I've mentioned a number of terms: responsible parties, applicable clinical trials. I'm not going to go into detail on these requirements today, and that's because my colleagues at CEDAR have 3 excellent on-demand webinars that you can review at your leisure. These provide a wealth of information. They provide both a general overview of the clinicaltrials.gov databank, walk through relevant definitions, laws, and regulations, and really provide an overview for what's needed to comply with the clinicaltrials.gov registration and results information submission requirements. Instead, I want to turn now and dive into the FDA requirements for clinical research and for clinical investigators in particular. FDA's legal framework starts with the Federal Food, Drug, and Cosmetic Act. So Section 505I in the FD&C Act is the statutory authority for FDA's oversight of clinical investigations to test safety and effectiveness of drugs. So the Code of Federal Regulations, or CFR, includes regulations describing this authority over the conduct of clinical investigations. The FDA's portion of the CFR interprets the Federal Food, Drug, and Cosmetic Act and related statutes. Section 21 of the CFR contains most of the regulations pertaining to food and drugs, and the regulations that are issued under Section 505I describe FDA's authority over the conduct of clinical investigations. Guidance documents represent the agency's current thinking on a particular subject. Because guidances are not regulations or laws, they're advisory only and generally not enforceable. An alternative approach to what's described in guidance may be used if such approach satisfies the requirements of the applicable statute, regulations, or both. So this is the overall legal framework in which clinical investigations are designed, conducted, overseen, and reported. FDA expects clinical investigators that are operating in this framework to know the relevant regulations, to understand their responsibilities, and to adhere to the regulatory requirements. FDA's regulations are intended to ensure the integrity of clinical trial data on which product approvals are based, and they're intended to help protect the rights, safety, and welfare of those who participate in trials. Key for clinical investigators are requirements relating to providing financial disclosure, related to obtaining legally effective informed consent from participants, ensuring the initial and ongoing review of the study by an institutional review board, and lastly, understanding the investigator responsibilities laid out in parts 312 and 812 for drugs and devices, respectively. These investigator responsibilities are also laid out in the Statement of Investigator, our Form FDA 1572. Our regulations provide that no investigator may participate in an investigation until he or she provides the sponsor with a completed, signed Statement of Investigator. If you look at the 1572, Section 9 outlines 8 commitments that I would encourage you to read closely before signing. Don't treat this like a EULA for an app. Each of these highlights a key expectation of the agency. In signing the document, you're agreeing to adhere to the IRB-approved protocol, with limited exception. You're also agreeing to make sure potential participants are appropriately consented to the study in accordance with our human subject protection regulations at Part 50, and in particular, that they're aware the drugs are being used for investigational purposes. And you're committing to maintain adequate, accurate records of the trial, among other things. So these obligations all center on the clinical investigator. Under our regulations, the clinical investigator is in charge and held accountable for study conduct, including for activities that he or she may delegate to members of study staff. So that really underpins the need to make sure staff and associates supporting trial conduct are aware of their obligations. Because while our regulations permit sponsors to formally transfer their responsibilities, for example, for study monitoring, to contract research organizations, clinical investigators cannot similarly transfer their responsibilities to sub-investigators, site staff, study staff, or to CROs. Because the investigator's obligation to personally conduct or supervise a clinical investigation is so critical to trial quality, FDA put out guidance in 2009 with our recommendations on how clinical investigators can demonstrate this oversight and avoid challenges. For example, most clinical or medical tasks require formal medical training and may also have licensing or certification requirements. Licensing requirements may also vary by jurisdiction, for example, by state or by country. Investigators need to take into account such qualifications or licensing requirements when considering delegation of specific tasks. In all cases, a qualified physician or dentist should be responsible for all trial-related medical decisions or care. During inspections of sites, there are cases where FDA has identified instances in which study tasks, like a physical exam or review of eligibility criteria, have been delegated to individuals lacking appropriate qualifications. Another area of focus in this guidance is supervision. So in thinking about whether to take on a trial, as a clinical investigator, you need to think about whether you have sufficient time to properly conduct and supervise the trial, taking into account the experience of your staff and their workload, the complexity of the trial design, and other ongoing trials that may compete for attention, among other factors. So I think this particular guidance can be a useful resource for thinking about how to put in effect your responsibility for supervising the clinical trial. I know Leonard and John covered decentralized trials yesterday, but I think it's important to revisit here since we're focusing on the investigator role and expectations for oversight. So as you heard, FDA issued draft guidance earlier this year on DCTs. So trials with decentralized elements are important to expanding the tent of research. Bringing trial activities to where participants are, to me, is among the promising strategies for reducing barriers to participation in clinical trials that can arise for participants who have to be at a specific facility for every trial visit. So that can be really limiting for those who have accessibility issues, those who don't live near a trial site, and for those whose family and job burdens preclude such on-site visits with the frequency required by a trial protocol. DCTs may include the use of telemedicine to conduct remote trial visits, the use of informed consent in an electronic format, and direct distribution of investigational product to patients. All of those may enhance trial convenience for participants, reduce burden on them or their caregivers, and potentially improve participant engagement. So I think decentralized trials really have the potential to be very beneficial. When thinking about trial oversight, the responsibilities of a clinical investigator conducting a decentralized trial doesn't differ from their responsibilities if the trial were a traditional site-based one. That is, the investigator still needs to comply with the IND application regulations found in Part 312 of the Code of Federal Regulations. And in general, the investigator remains responsible for the overall conduct of a decentralized trial, including oversight of individuals that are delegated to perform trial-related activities. A key difference for decentralized trials is the extent to which different decentralized elements are involved. So the extent to which the investigator is using telehealth, using trial personnel working remotely, digital health technology, or incorporating local healthcare providers into the conduct of the trial. So just as a reminder, as defined in the draft Decentralized Trials Guidance, local healthcare providers are individuals who are carrying out trial-related services that are part of their routine clinical practice, like taking vitals or conducting a physical exam. And according to the draft guidance, to be considered a local HCP, these services shouldn't require having a specific, detailed knowledge of the protocol, the investigator's brochure, or the investigational product. So when considering a decentralized trial and considering the extent of decentralized elements included, one of the things that the investigator should think through is his or her capacity to oversee participants' safety, depending on how many personnel are remote, whether there are local HCPs involved, and the extent to which telehealth might be involved, for example. So based upon these differences and the decentralized features of the trial, including things like additional training, coordination, and or standard operating procedures might be necessary to ensure consistent implementation. To me, much of this comes down to what's been advocated in ICH E6, upfront risk assessment and management, in thinking prospectively through challenges that might arise in implementing the trial. That is, thinking about what makes sense for this trial for this planned patient population. For example, if there's a digital health technology involved, is there any specific training needed for trial personnel, participants, or for both personnel and participants? If a DHT is used, are they available and suitable for all participants? You might also think through if there are adverse events identified remotely, how will these be reported and managed? So again, to me, there's an aspect of applying what we already know works and really applying risk-based approaches to implementation and oversight. For decentralized trials and for traditional site-based trials, investigators conducting drug trials have to complete Form 1572, the statement of the investigator. In addition to outlining the investigator responsibilities that we've been discussing and will continue discussing, the 1572 includes information like the name and address of the clinical investigator, the names and addresses of research facilities where the trial will be conducted, for example, medical schools or hospitals. It includes the names and addresses of clinical laboratory facilities that are to be used in the study, and the names of any sub-investigators. So a question that often comes up about whether trial personnel should be included as sub-investigators on the 1572. That's just a question we often get. So when trial personnel contribute directly and significantly to the trial data, they should be included as sub-investigators on the 1572. So if someone's assessing an endpoint, determining eligibility, or adjudicating adverse events, they would typically be making a direct and substantial contribution to the trial data. Local healthcare providers, as defined in the draft DCT guidance, should not be listed on Form FDA 1572 as sub-investigators. Local HCPs should, however, be included in a task log, which the draft guidance discusses in more detail. If you're looking for further recommendations just generally on completing a 1572, there is an information sheet, and you can see at the bottom of the screen, that provides guidance for sponsors, clinical investigators, and IRBs. So it walks through some frequently asked questions, and this was a guidance that we published in May of 2010. So just to touch quickly on device trials, similar to investigators and drug trials, investigators and device trials should also list local HCPs in a task log. And separately, sponsors of device trials must provide a list of all investigators as part of their IDE application. So let's come to our first challenge question and make sure, despite the fact that we are at least on East Coast time, having this discussion shortly after what would be lunchtime. Let's make sure everyone is still with us with a challenge question. So, per the draft decentralized trial guidance, where should local healthcare providers be listed? Should they be listed in the Form 1572 as a sub-investigator? Or B, should they be listed on the 1572 and in a delegation log? Perhaps it's C. Local healthcare providers should be listed in the task log. Or is it D, the 1572 and the task log? I'll give you a moment to peruse, think about it, and hopefully everyone in the audience has come up with answer C, the task log. So this task log is a concept described in the draft DCT guidance, and that is where we recommend that any local healthcare providers as defined in the draft guidance be listed. So let's turn to some key investigator responsibilities for safety reporting in clinical trials. In September of 2021, we issued a draft guidance on this very topic. So as an investigator, your responsibilities include monitoring for the occurrence of adverse events in study participants that you've enrolled in the trial and evaluating whether events are serious or not. Serious adverse events, or SAEs, need to be reported immediately to the sponsor. Non-serious adverse events should be recorded in trial records and reported to the sponsors according to the protocol requirements. As an investigator, you also have an obligation to review IND safety reports that the sponsor may send you. FDA generally considers that serious and unexpected adverse events that meet the criteria for an IND safety report, we generally consider those to be unanticipated problems involving risks to human subjects or others. And under our regulations, as an investigator, you must report such unanticipated problems to your IRB. These responsibilities don't function in a vacuum. For safety reporting to work as intended under our regulations, the sponsor, the IRB, and the investigator must each fulfill their responsibilities, and they need to frequently communicate with each other regarding the evolving safety profile of the investigational drug. This is what you see on the screen is a very basic, high-level generalization showing how this continuous process should work for IND safety reporting. So let's walk through it. So the sponsor must evaluate SAEs and submit IND safety reports to the FDA and to all participating investigators. The sponsor must also evaluate safety signals in the aggregate Where appropriate, the sponsor needs to modify the investigator brochure, the protocol, and/or the informed consent based on the evolving safety signals of the investigational product. For the sponsor to be able to comply with these requirements, the investigator has to identify adverse events, determine if they're serious, and report them to the sponsor. Further, the investigator has a continuing duty to protect the safety of trial participants. To appropriately do so, they need to review the IND safety reports that the sponsor sends to them. In turn, the investigator must report unanticipated problems, including those that arise from IND safety reports, to the IRB. Where necessary, the investigator may need to modify the informed consent form and submit it to the IRB for review and approval. The IRB must then comply with its obligation to continuously review the research by reviewing unanticipated problem reports from the investigator. The IRB must follow its written procedures to ensure the safety and well-being of participants. So I want to quickly move us to a second challenge question. So drum roll, please. Who has responsibility for reporting unanticipated problems involving risks to human subjects or others to the IRB? Is it A, the sponsor, B, the investigator, C, the contract research organization that's supporting the sponsor, or D, all of the above? Well, we did talk about everyone playing a role in safety reporting. I hope you'll recall that it is B, the investigator, who has the responsibility for identifying and reporting unanticipated problems involving risk to human subjects or others to the IRB. We just mentioned that new safety information can lead to changes in the informed consent document. So let's go back to the beginning of the study and talk a little bit about the requirements for informed consent in our regulations. First, under 312.60, unless certain exceptions are met, such as the exemption from informed consent requirements for emergency research at Part 5024, the investigator must obtain the informed consent of each participant to whom the drug is administered. Informed consent needs to meet the requirements for required content that's outlined in 21 CFR Part 50.25. There are 8 basic elements of informed consent that I'll walk through briefly. There are also other elements that might be necessary depending on the particular study. But the basic elements are that the study involves research, an explanation of the purpose of the research and the expected duration of the participation of a trial subject, a description of the procedures to be followed, and identification of procedures which are experimental. Second, there needs to be a description of any reasonably foreseeable risks or discomforts to the subject. There also needs to be a description of any benefits to the subject or to others which may reasonably be expected from the research. There also needs to be disclosure of appropriate alternative procedures or courses of treatment, if any, that might be advantageous to the subject, as well as a statement describing the extent, if any, to which confidentiality of the records identifying the subject will be maintained. And this statement needs to note the possibility that the Food and Drug Administration may inspect the records. For research involving more than minimal risk, the consent should include an explanation as to whether any compensation and an explanation of whether any medical treatments are available if injury occurs, and if so, what they consist of, or where further information may be obtained about it. There needs to be an explanation of whom to contact for answers to pertinent questions about the research and research subjects' rights, and whom to contact in the event of a research-related injury to the participant. And last, the mandatory elements include a statement that participation is voluntary, that refusal to participate will involve no penalty or loss of benefits to which the participant is otherwise entitled, and that the participant may discontinue participation at any time without penalty or loss of benefits to which they're otherwise entitled. So those are the required elements. One thing that I would emphasize is that those required elements should be kind of described in a participant-friendly language that they're able to understand. I think sometimes we see consent forms that are quite voluminous that may be impenetrable to those considering participating in a trial. So, keep that in mind as well. There's information that needs to be conveyed, but it can be conveyed in a manner that helps the subject, the potential participant, understand whether a particular trial is a good option for them. And so we've been talking about the document, but the informed consent process needs to also provide an opportunity for study participants to ask questions and receive answers. And process is a really important word. Informed consent is just not the document or a signature on a document. It's an ongoing process that must occur before any study-related procedures or tests are conducted, and includes the process extends into trial conduct and making sure that participants are made aware of any new information that might change their decision to continue participating in a particular trial. When thinking about obtaining informed consent, we often get the question of whether the investigator has to sign the informed consent document. So the answer is no. If you're thinking about a requirement that the person conducting the informed consent discussion must sign and date the document, you'll find that in the ICH E6 guideline, but not in FDA's regulations. So if you have questions about consent, we published a final guidance on informed consent, providing guidance for IRBs, for clinical investigators, and sponsors. So this is relatively hot off the press. It published just a few months ago in August. It has a wealth of information about the informed consent document, the informed consent process, and other ethical considerations. It's organized to provide, in the first part, general guidance on FDA's regulatory requirements for informed consent. And that's followed by a discussion of the roles of various parties for consent. And finally, it includes a series of frequently asked questions about informed consent. So if you have a question, it might possibly be answered, or hopefully be answered in this guidance. If it's not, I would encourage you to send your questions to our GCP mailbox at gcpquestions@fda.hhs.gov. We published a guidance in 2016 that's specific to the use of electronic informed consent. So this was a joint guidance that we issued with the Office of Human Research Protections, the office that governs human research protections for federally funded and supported research. So this particular guidance provides recommendations on the use of various electronic media to obtain and to document informed consent. And by aligning with advances in technology, we think that electronic informed consent may provide some advantages. It may improve participants' comprehension, and it may also increase their engagement. It allows the opportunity for study participants to be able to review informed consent forms and to sign them from the comfort of their own home or wherever is most convenient for them. So from the patient perspective, this might translate into convenience. They don't have to travel to a research site for the purpose of informed consent. They can review the consent form and consult with their family members without having the pressure of time or the constraints of a particular on-site visit. Participants who have the opportunity to review the consent form at their leisure may also have time to make arguably a more informed decision. And this could include not just electronic informed consent, but supplementing consent forms with electronic technologies that can really help participants better understand the research. And these technologies may be capable of engaging potential participants more than a traditional paper consent form might. So for the last part of my time with you, I want to turn to some proactive practices. Prevention always being better than remediation when it comes to trial quality. If you're a clinical investigator conducting a trial for a sponsor, there are approaches that sites have taken to kind of avoid being non-compliant with the protocol. The first is to appraise the study beyond just assessing whether it's of scientific interest to you, but really looking at how the study as written might translate to operations at your site. What are the anticipated visits? What's the participant flow through these visits? And is there a difference from the standard clinic workflow for similar encounters? Are there telephone visits that wouldn't normally occur that need to be accounted for in your workflow? Second, you could consider what data activities are really critical to conduct and collect, and are these part of the normal clinical encounter, or again, something to plan for? You might consider whether there'll be specialized expertise needed, for example, a radiologist for certain scans, or any special equipment needed for the study. I really think the sites that have used it successfully are really looking for those as well as other study-specific nuances that may differ from other trials, like adverse events of special interest that aren't SAEs but need to be reported differently than other adverse events, for example, on a special form or on a different timelines. Some sites may do dry runs or beta tests to double-check whether there's clarity on roles and responsibilities, whether there's sufficient staffing for visits that are particularly activity-laden, and to make sure that they have clarity on how and where source data will be recorded. Once the study is up and running, I think there's also opportunity to look at records from study visits in near real time to help identify any gaps or departures early so that actions can be implemented to prevent them from recurring and becoming persistent. For those sponsor investigators in the crowd who are designing trials, a very important proactive practice is described in ICH E8 Revision 1, and that's the concept of clinical quality by design, of focusing on the critical aspects of the trial, the things that must go right to yield credible conclusions and reliable data and to protect trial participants. Quality by design means focusing on these critical aspects at the time of design. And this is a concept that is carried through into trial execution and ICH E6 R3. So why at the time of design? Well, we know that clinical trials are a real fundamental part of research. Their results are essential for evidence-based healthcare decisions. And if we don't address design issues, we risk yielding unreliable and/or inadequate evidence. A poorly designed trial, in addition to a poorly conducted one, wastes resources and may not align with ethical principles. So when you're designing a trial, you have an opportunity to avoid these challenges. Design is the point when you can identify critical aspects of trial design and take steps by tailoring design to avoid the kinds of errors that impact regulatory decision-making. That is, those that undermine participant safety or those errors that challenge data reliability. Protocol design and development is also an opportunity to ask, why have we included this, whether it be data collected or a procedure conducted? It's an opportunity to ask that question and then to streamline the trial where feasible. It's also a last checkpoint to verify that the trial that you've designed will answer that important scientific question you had in mind when starting out. And finally, the outcomes of a protocol design stage review can translate to efficient, focused implementation and oversight. The risk-based quality management and monitoring that's in FDA's guidance in ICH E6 R3 and in guidance from other regulatory authorities. Let's walk through an example of how this could help both sponsor and investigator to proactively assess a protocol before starting a study. Again, this case is thanks to my CEDAR Office of Scientific Investigation colleagues. It relates to a marketing submission that included a very large randomized cardiovascular outcome study powered based on the predicted number of primary endpoints. The primary endpoint was MACE, so major adverse cardiovascular events, and there was an independent adjudication committee for MACE events. During application review, FDA identified sites for inspections, including a site, we'll call it Site A, that had high enrollment and data trends suggesting potential underreporting of events, both safety events and for the primary endpoint. On inspection, the site records showed events that appeared consistent with a MACE event, the primary endpoint. These events had been sent to the adjudication committee, but had been adjudicated negatively. The challenge, according to the PI in discussions during the inspection, was that when events occurred outside of the site and its network, they had extreme difficulty obtaining any records that were needed by and requested by the adjudication committee due to a reported litigious climate. So in the absence of records to confirm these events, the impact was that the predicted number of primary endpoint events was not reached. the study was underpowered and the sponsor had to, at substantially increased cost and with an increased trial length, they had to implement a protocol amendment with a re-estimated sample size. So as I think about this, the sponsor might have considered global differences in site practices that might have revealed this as a risk requiring some mitigation. For example, by considering how and where the endpoint might occur, be documented, and how the documents needed by the adjudication committee would be obtained. The site, in a dry run or walkthrough of the processes, might have identified the need to obtain documents from external institutions as a potential challenge. So now we've come to the end of our somewhat whirlwind tour. So I'll summarize by highlighting the value and the role of clinical investigators in delivering trial quality. And for those sponsor investigators taking on the dual role of clinical investigator and sponsor, you also have the additional obligation of transparent reporting of results of applicable clinical trials to the clinicaltrials.gov databank. Whether you are an investigator or a sponsor investigator, you have an obligation to make sure that staff supporting trial conduct have a clear understanding of the protocol and their responsibilities under FDA regulations. At stake at the end of the day is public confidence. We all suffer if the public loses confidence in trial integrity. Conversely, trials that are designed well, conducted well, and that respect participants and that are reported transparently build public confidence and encourage broad participation, which ultimately yields safe and effective products. So I think we have time for questions later this afternoon. Happy to address them. And again, just a reminder, you'll see the email at the bottom of the screen. That is our, within my office, we maintain an inbox for GCP questions, and that is gcpquestions@fda.hhs.gov. I probably should have made that a challenge question because I hope it's something you remember as a resource. If you're looking for things in guidance, can't find it, have questions that aren't addressed in guidance, it's a very valuable resource. But with that, I will close and look forward to answering your questions later. Thanks so much.
Thank you for that introduction. Good afternoon, everyone. Today, I will be talking with you all about clinical investigator site inspections and what to expect. This presentation will build upon the talk before me on investigator responsibilities, so kudos to the organizers for making such a thoughtful schedule. Now that you know your regulatory responsibilities as clinical investigators, we are now going to look at them from an inspections perspective. We will discuss the purpose and focus of clinical investigator inspections, describe the inspection timeline and activities, and lastly discuss potential enforcement actions if significant regulatory violations are identified through the inspections. In terms of regulatory responsibilities as CIs, this list is not inclusive, but probably includes those most relevant to inspections. As CIs, you are responsible for ensuring that the investigation is conducted according to the investigational plan, the protocol, and applicable regulations. You are responsible for protecting the rights, safety, and welfare of human subjects participating in your trials. You're responsible for obtaining informed consent and IRB approval or oversight, and for maintaining adequate control of drugs or any other FDA-regulated investigational product under investigation. Clinical investigator inspections fall under the umbrella of good clinical practice inspections. Good clinical practice, or GCP, is an international ethical and scientific quality standard for the design, conduct, monitoring, auditing, recording, analysis, and reporting of clinical trials. So these inspections evaluate clinical trials for compliance with GCP and regulations, so as to ensure the rights, safety, and welfare of human subjects, as well as ensure the reliability of the safety and efficacy data submitted to FDA in marketing applications. Other inspected clinical trial entities include sponsors, contract research organizations, and institutional review boards. Clinical investigator inspections will look at how the trial was conducted at your site. And so the first question the inspection will attempt to answer is, was the study conducted according to the protocol? The items on this list are just some of the areas we will be looking at with respect to protocol compliance. For example, did subjects fulfill all inclusion criteria and none of the exclusion criteria? Were randomization and blinding procedures followed? Did anyone become unblinded? What was the data flow from your site to the sponsor's database? Were the pre-specified study visits, procedures, and evaluations performed as written in the protocol? And were subjects dosed correctly? Another question we are trying to answer with the inspection related to trial conduct is, did the study conduct comply with regulations? Again, this list is not inclusive, but areas we will look at include adverse event reporting, IRB oversight, consent procedures, financial disclosures being submitted, electronic records being managed, and study drug accountability. Another important component of clinical investigator inspections is source data verification, where the data generated by your site is compared to the data that was submitted to FDA. Now, as you all know, the amount of data generated by a site in a given trial can be a lot, so we tend to focus on high-yield data such as primary endpoint data, whether adverse events or protocol deviations were unreported, and laboratory data. However, the specific data we will validate at your site will be tailored based on the FDA review team's interests. There are two types of clinical investigator inspections. The first type is routine surveillance inspections, and these coincide with a pre-marketing application review, in that the sponsor has submitted an application to market a product, and clinical trials were used to support that application, which now need to be inspected. These inspections are generally pre-announced. We let you know that we are coming. The other type of inspection is for-cause or directed inspection, where we investigate potential regulatory violations. These stem from a complaint that we have received. Sponsors, IRB, study staff, study participants, virtually anyone can submit a complaint to FDA. These for-cause or directed inspections are generally unannounced. They are unannounced because if FDA is going to inspect you for a potential regulatory violation, we don't want to give you time to fix it before we get there. For logistical reasons, foreign inspections are generally announced. First challenge question: which is not a regulatory responsibility of clinical investigators? A, obtaining informed consent. B, obtaining IRB review or approval. C, paying subjects for trial participation, or D, ensuring that the investigation is conducted according to the investigational protocol. So take a few seconds to consider which answer you think is correct. And hopefully you thought it was C, paying subjects for trial participation. All the other options are your regulatory responsibilities. Probably the section you've most been anticipating, what exactly happens during an inspection? Before the inspection, there is the pre-announcement, again, if this is a routine surveillance inspection. You will be contacted up to 5 days before the start of the inspection. There will be some discussion of inspection scope and logistics, such as the protocols being covered, and the records we plan to look at, just in case you need to retrieve them or gain computer access, how long we anticipate being there, again, anticipate, based on the number of subjects and protocols the inspection will cover, and the workspace. On the very first day will be the opening meeting. The FDA team usually consists of one or more investigators from the Office of Regulatory Affairs. Their other title is known as Consumer Safety Officer. On occasion, a subject matter expert from the center having jurisdiction over the product being researched may also be in attendance. Regardless, whoever is representing FDA will provide their credentials and a notice of inspection to you. At this opening meeting, we will discuss the basis and scope of the inspection, such as the number of studies, subjects, and data to be verified, your availability, and the staff's availability for meetings or questions as they come up throughout the course of the inspection. And as part of the inspection process, we may collect exhibits. So we may ask you to provide specific documents electronically for us to take. And so we will discuss with you how you will provide those documents to us, usually through scanning. There will also be an interview just to ascertain your knowledge of the study or studies being inspected. Questions could include how you were selected by the sponsor, asking you to provide an overview of the study at the site, describing how subjects were recruited, whether any significant issues were experienced, such as protocol deviations or serious adverse events, and how these were handled, and a list of studies conducted by yourself. This gives us an idea of your workload, not just clinically, but as a researcher. We may also do a facility tour of the various areas at the study site, such as how you stored the investigational medical product. However, the bulk of the inspection will be spent reviewing records, so tidy, well-organized, and comprehensive record keeping is key. In terms of study records, we will look at protocols, correspondence from the IRB or the sponsor, monitoring reports, signed investigator statements and financial disclosure forms from you and your sub-investigators, training logs, how you delegated tasks to your staff, laboratory certifications, and medical product accountability records to your site, such as shipping and receipt records. In terms of subject-specific records, we will look at informed consent forms, visit worksheets, laboratory reports, progress notes, adverse event reports, efficacy endpoint assessments, and medical product accountability records on a subject level, such as dosing and administration. Generally documents that show subject participation at your site. On the last day of our inspection, there will be an inspection closeout meeting where we will provide a summary of inspection findings and discuss any issues of concern. If any of the issues raise concern for possible deviations from federal regulations, a Form 483 will be issued. Note the word possible, as back at FDA, after we have reviewed everything, we will determine whether each observation constitutes a regulatory violation, and if so, determines the impact on data integrity and subject. If issues or observations are discussed at your closeout meeting, you are welcome to provide a verbal response at the meeting, or you may provide a written response. This written response is optional, but it should be received by FDA within 15 business days of inspection close. Although it is optional, a written response may be helpful in that it demonstrates to FDA your acknowledgement and understanding of the observations. It demonstrates your commitment to correct the observation to FDA, and probably most importantly, it may be considered in the final FDA compliance decision. If you do wish to provide a response, it should contain the following: a corrective and preventive action plan for each observation, a timeline for completion of actions, a method for verifying and monitoring the effectiveness of the actions, and submitting documentation such as training records or standard operating procedures that you have created. The final FDA classification will occur after formal review of the inspection report, 483, and the written response if provided. The basis for the final FDA classification is whether objectionable conditions were observed. Objectionable conditions are those where subjects were exposed to unreasonable and significant risk of injury, subjects' rights, safety, and welfare were compromised, or data integrity and reliability were compromised. With those criteria for objectionable conditions in mind, let's move on to the types of FDA classifications for CI inspections. The first is no action indicated, or NAI. No objectionable conditions or practices or the significance of the documented objectionable conditions found does not justify further action. For this, you will receive a letter. Regarding the timing of the letter may be released to coincide with other inspection letters or the application review, so it may take a while for you to receive the letter after the inspection, but it will eventually come, don't worry. The next classification is voluntary action indicated, or VAI. Objectionable conditions were found and documented, but the center is not prepared to take or recommend regulatory action since the objectionable conditions do not meet the threshold for regulatory action. For this, you will also receive a letter, but it will just describe those objectionable conditions. The final FDA classification is official action indicated. Objectionable conditions were found and regulatory action is recommended. Before we move on to the actual outcomes of an OAI, let's look at some common inspection findings that might be included in the Form 483 or inspection letter. So here's a list of regulatory violations and the corresponding regulation. So first, the investigation may not have been conducted in accordance with the investigational plan or the signed investigator statement. Failure to prepare or maintain accurate case histories, so poor record keeping. Failure to obtain informed consent in accordance with 21 CFR Part 50. Investigational drug disposition records were not adequate. And failure to report promptly to the IRB all unanticipated problems involving risks to subjects. And failure to report adverse events promptly to the sponsor. So back to OAI. Some outcomes could include a letter describing those objectionable conditions, or disqualification, wherein the investigator is not able to conduct clinical investigations involving products regulated by FDA. Other enforcement actions could include civil money penalties or debarment. Debarment usually follows after a federal felony conviction, and in that situation, the individual may not provide services in any capacity to a drug company. Okay, challenge question number 2. Which of the following statements is not true? A written response to a 483 is required. B, a written response to a 483 is due within 15 business days of inspection close. C, outcomes of an OAI classification could include disqualification or a letter. Or D, a written response may be considered in the final FDA compliance decision. So take a few seconds to consider which answer you think is correct. And hopefully you thought it was A written response to a 483 is required. It is not required. It is optional. But if you do choose to do so, it is due within 15 business days of inspection close, and it's possible that it may be considered in the final FDA compliance decision. If you want more information about what exactly goes on in a clinical investigator inspection, you can check out this compliance manual, which lays out all the items to be covered in an inspection. So, building on your knowledge of your regulatory responsibilities as CIs, in this talk we have discussed the purpose and focus of CI inspections and what exactly goes on during an inspection. And so hopefully now that you know what to expect, you can be prepared if an inspection occurs. The best way to survive an inspection is to always be prepared for one. That concludes my presentation. I will be back shortly to answer questions. Thank you.
Thank you for joining this session. My name is Kasa Yalo. I'm the Director of the Division of Clinical Compliance Evaluation in the Office of Scientific Investigations within the Center for Drug Evaluation and Research at the FDA. My presentation is intended to provide you information and insight on international clinical trials from GCP perspective. Disclaimer, the views expressed in this presentation are those of mine. They do not necessarily represent those of the Food and Drug Administration. My presentation has two objectives, and first and foremost, it is intended to provide you information on a general overview of international clinical trials from GCP perspective. Secondly, it's also to provide you information on how to comply with FDA's regulatory requirements when you are engaged in conducting clinical trials internationally. As it is well known, clinical trials are increasingly global. As a result, data regulators such as FDA use to authorize drug products often are generated from trials that are conducted across the globe. Clinical trials in support. As a result, data regulators such as FDA use to authorize drug products, you would be required to ensure IRB review, approval, and reporting requirements are met. And in addition, as investigators for IND studies, you would be required to ensure FDA has access to inspect your records and reports as applicable or appropriate. And IND studies investigators will be also required to ensure the informed consent is adequately obtained. And last but not least, and investigators are specific procedures, and they may also review the informed consent and the IRB oversight process, as well as documentation to determine the ethical development of the product at the specific site. If the inspectors may also compare line listings generated from a sponsor submission with the source documents to verify the reliability of data submitted to regulators. And if the inspectors may review the study participant enrollment process, and they may also review the monitoring activities at a particular site. Inspectors may also review clinical investigators' compliance with applicable regulatory expectation when it comes to reporting, such as safety reporting, financial disclosure reporting. And they also may look into test article control at the site, and they may review records custody and retention. Sponsor and CRO inspection focus. Sponsor and CRO inspection generally focus on key areas such as reviewing protocol compliance, data integrity, regulatory adherence, and overall quality of the clinical trial conduct. The inspection includes reviewing organizational structure, registration of trials according to a regulatory requirement, assessing the investigator selection process, and evaluation of the adequacy of communication with clinical investigators. The inspection also review the adequacy of monitoring process, the quality audit procedures, and adverse event reporting. The other areas of focus include assessing data handling and the control of investigational product by the sponsor and CRO. The inspection of CRO and sponsors also review computerized processes, electronic records and signatures, and adequacy of record keeping as well as record retention. It also reviews the adequacy of financial disclosure reporting. Like during the inspection of clinical investigators, the inspection of sponsor and CRO review if a clinical trial is conducted in accordance with good clinical practice. Before we conclude this presentation, I would like to share the quote from FDA's Principal Deputy Commissioner Janet Woodcock as take-home point. Wherever clinical trials are conducted, it is important to have drug development programs that reliably produce high-quality data acquired in a manner that will not jeopardize the rights, safety, or welfare of trial participants. Thank you for your attention.
Hello, everyone, and we're moving into our final question and answer period of the day. Thank you so much to the three speakers. I think, as indicated in the number of questions that we're getting, you covered very practical information that's relevant to the clinical investigator and in the day-to-day role of the clinical investigator. Very, very important role in the clinical trial space. I'd like to start the Q&A session. Stephanie, you're hopefully still on the line. So question for you, how long do I have to keep records for?
Hi, thank you for that question. So FDA regulations require that you keep records for 2 years after the date a marketing application has been approved for the indication it's being investigated. However, you can retain records for a longer period based on the sponsor's procedures, the protocol, or your own site's standard operating procedures.
Great, thank you so much. And then a related question, is it acceptable to retain paper records as electronic records and destroy the original paper records to minimize storage requirements?
Hi, thank you for that question. Yes, it's acceptable, to help with storage, to convert paper documents into an electronic record, just provided that the electronic record is a certified copy of the original information, meaning that this certified copy or electronic record has been verified to have the same information as the original, either through a dated signature or some sort of validated process to make sure that it's the same. It's great if you have written standard operating procedures, that describe how you're going to make these copies, how you verify that they are like the original, and document that.
Great, thank you so much. And clearly record keeping is very central of importance to investigators. We have another question on that. If the inspection identifies issues with record keeping or protocol compliance that were caused by research staff or sub-investigators, who is responsible?
Yes, thank you for that question. As Anne actually talked about, as clinical investigators, you all have ultimate responsibility for study conduct at your site. So if there are certain, if you get delegated certain tasks to other individuals, and that there have been issues related to their conduct, you are responsible for them, and so that's why the inspection is tied to the clinical investigator.
Great, thank you so much. Anne, several questions for you related to the 1572 forms. So one, does each sub-investigator need to sign a separate 1572?
Thanks. That's a great question. The 1572 is actually signed by the clinical investigator. So some might call that the principal investigator. So the sub-investigators don't individually need to sign a 1572. Their names are actually listed on the 1572, but they don't need to complete one. Thank you.
And then I think a slightly different but related question, do we need to resubmit the Form 1572 if a sub-investigator is added or retired during a clinical trial, or do we only need to resubmit Form 1572 when there is a change in the principal investigator?
That is another great question, and one that I think, I think has been an area reasons when a 1572 needs to be completed. And those are when there is a new clinical investigator or when the clinical investigator at a particular site is replaced. So you don't need to complete an updated 1572 if there are changes to sub-investigators. Those can be documented in study records and communicated to the sponsor, and they can update the IND as appropriate.
Thank you. And one more while we're on the subject of 1572s. You mentioned the Form 1572. What do I do if I'm an investigator outside the United States in a country where I am not allowed to sign such a document?
Thank you. Another common question, and one where we actually have a full guidance on the topic. So that on 1572s that includes this topic, I should say. So if a clinical study is conducted at a foreign site under an IND, all FDA IND regulations, including the requirement to obtain a signed 1572, have to be met unless the sponsor requests and is granted a waiver that provides for specific exceptions. So in the case where a clinical investigator in a foreign country can't or won't sign the 1572, for example, because regional, national, or local laws or regulations prohibit signing it, the sponsor may submit a request for a waiver of the 1572 signature requirement, and that's under our regulations at 312.10. So waiver of the signature on the form 1572, when granted, enables the study at foreign sites to be and or remain under an IND, even though the investigator at those sites can't sign the form. And that's provided that the sponsor complies with the terms of the waiver. So thank you.
Thank you for that. And while we're on the subject of non-US sites, CASA, we have a few questions for you. Is FDA's approach to inspecting non-US sites or clinical trials different from its approach to inspecting sites or clinical trials in the United States?
Thank you for the question. I think that's a great question. And can you hear me? Just want to make sure. And
yes.
The inspection approach is the same, irrespective of the location where clinical studies may be conducted. the approach of the inspection would be the same, and also the selection of sites for inspection would take the several factors that are relevant to both U.S. and non-U.S. sites or clinical studies that are conducted in the U.S. and outside U.S. And we take a risk-based approach to identify clinical investigator sites for inspection. And that takes into account the application label factors, whether the application is for new molecular entity, or if the study in support of the marketing application is a very complex study, or the study that we would like to inspect and to understand better the conduct of the study. And also, of course, the site level factors, and taking into account the comparative performance of the site level information, whether it's efficacy or safety, and also the compliance history of the sites. And the bottom line, our inspection approach for US and non-US sites are the same. Thank you.
Terrific. And I think your answer actually covered the next question on the list as well, which is what are the selection criteria for foreign sites inspections or clinical trials? So if you have more to add, please do there. Otherwise, the next question for you is there a difference between US and non-US based investigators in terms of the types of deficiencies noted during inspections?
Great, yeah, thank you. I'll try to address both. And as you said, I touched upon some of the things that were asked. And we, in general, taking into account the resource issue, also the breadth of the clinical investigator sites across the globe, we have several things that to take into account before sending someone to inspect sites outside US. And we, in general, tend to send international inspections when the internationals, when there are insufficient domestic data to provide us confidence on reliability of the information, as well as making sure that the study subjects were protected during the clinical study. Also, we take into account, rarely close to 10% of the data sometimes comes solely from foreign countries, and those instances make us to go to do an inspection outside US. And if you do see conflicting data between the US, the data that came or generated from the US sites versus outside US, of course, we'll be doing an inspection outside US, as well as scientific misconduct based on compliant, based on information that we get. If they indicate that the foreign sites may have been involved within that kind of scenario, we do send our inspectors to conduct inspection. And the second question you asked me was about the types of findings between US and non-US sites. In general, they are very much similar. And often the most common, I think, during the talk, Anne or Stephanie may have, I think Stephanie raised about that. And often we see investigators failing to follow the investigational plan and the protocol. That's the most common finding, or the type of inspectional findings we come across. And keeping the accurate records is another challenge that sometimes we come across. And the inadequate drug accountability, as you all know by now, our citations are based on our regulatory requirements. And generally, failure to follow the investigational plan and the protocol, and maintaining accurate study records, and as well as adequate drug accountability are the most common causes of inspectional findings, the most common inspectional findings we identify both in the US and outside US. Thank you.
Great, thank you, Casa. Continuing on the theme of inspections, Stephanie, a couple of questions for you. The first is, in addition to the clinical investigator inspection, would FDA perform a GCP inspection at the CRO, Clinical Research Organization, and or the sponsor sites during the BLA NDA pre-market application review?
Hi, thank you for that question. Yes, although the topic of this session was clinical investigators, yes, we do inspect contract research organizations and the sponsor for marketing application review.
Great, thanks. And one additional question there. What guidance are you able to provide a sponsor who may want to request a site inspection? How often does this happen, and what types of concerns would prompt a sponsor to request an inspection?
Hi, thanks for that interesting question. Sponsors don't necessarily request FDA outright to perform site inspections. Sponsors can perform monitoring visits and audits or obtain audits, but what they can do is inform FDA of issues with their particular site, and that can trigger an inspection with FDA. And so sponsors can let FDA know via an email box for good clinical practice issues through the IND submission application, or if there's a marketing application, they can include issues with particular sites in the clinical study report or in other sections in the marketing application. And so those are methods that FDA can become aware of issues with a particular site. And some issues that we look seriously at or things are like sites being terminated for issues with subject safety, lack of clinical investigator oversight, or data integrity or fraud.
Thank you. And Anne, we have a couple of questions for you related to informed consent, which you also covered during your talk. The first is, does the pre-screening process also need to be conducted after the informed consent form is signed? Pre-screening and screening procedures to determine trial eligibility. Is signing required?
So that's a good question, and it's a topic that we have a guidance on screening tests prior to study enrollment. And that guidance highlights that procedures that are, any clinical screening procedures that are performed solely for the purpose of determining eligibility for research, you need to obtain informed consent prior to initiating those procedures. These clinical screening procedures that are for research eligibility are considered part of the subject selection and recruitment process, and therefore require IRB oversight. So thanks for that question.
Great, thanks. And another informed consent question. Can informed consent be provided by a family member if a trial therapeutic is indicated for trauma victims who frequently will present to the emergency department unconscious?
So another good question, and one that gets to the concept of a legally authorized representative and who can serve as a legally authorized representative and what their role is. So our regulations define a legally authorized representative, or LAR, as an individual or a judicial or other body authorized under applicable law to consent on behalf of a prospective subject to the subject's participation in the procedures involved in the research. So the key point here when thinking about who can consent on behalf of the subject as a legally authorized representative is that they have to be authorized under applicable law to consent on behalf of a prospective participant. So once identified, that legally authorized representative may sign the consent form for the prospective participant, should that individual not be capable of providing their own informed consent. And they also should be consulted to make decisions on behalf of the subject and to assure that any such decisions are in the subject's best interest while the participant is enrolled and continues within the study. You know, so I think that is hopefully that gets to the question. So thank you.
Yes, thank you, Anne. CASA, another record-keeping question. Many developing countries do not have the same record-keeping and data collection requirements as the United States. What precautions should U.S. sponsors or CROs use when conducting clinical trials internationally?
Wow, that's a great question. And yeah, that happened presently. I think it's sponsored and CRO. We need to, earlier Anne was talking about quality by design. We need to look into several factors before considering conducting a study outside the country where they want to submit marketing application. And as you all know, and during presentation also I mentioned about applicability of the population and the medical practice and the standard that may exist in certain countries need to be taken into account. Of course, and the sponsors are expected to ensure that the clinical study they're planning to conduct and the clinical investigators have the appropriate resources to meet the regulatory expectations. Therefore, I think it's very important to have a discussion with sponsors and CROs before one join to conduct clinical investigator as a clinical investigator a clinical trial. And it is a very difficult subject, but the sponsor should provide resources that would be necessary to conduct the clinical study and to ensure that data are reliable and appropriate for regulatory audit, and that the patients are safe and their rights and their safety and well-being are protected, and also to comply with the regulatory expectation, whether the U.S. regulatory expectation or outside. I think my advice to clinical investigators or to people who want to become clinical investigators, it's very important to assess the environment and work with the sponsors to obtain the necessary resources to maintain records. And I think the technological advancement would help countries like a third part of the world. Perhaps the cloud sources could also support in maintaining some of the records that may be needed in clinical trials. And the bottom line, it depends, and they need to discuss with the sponsor. Kim?
Great, thank you, Casa. In a more general question, what are the major mistakes to avoid when conducting clinical trials internationally?
Yeah, for the sponsors, I mentioned, and again, the acceptance of data by the US FDA depends if the data is applicable to the US population and also the medical practice here in the US. Therefore, it is very important the sponsors determine that's the case. And of course, earlier we were talking about GCP. Both Anne and Stephanie touched upon GCP, and they need to make sure that they have investigators, that they have the recognition or competence to conduct clinical studies, and also they need to make sure that regulators such as FDA would be able to audit the data when that deems necessary. Therefore, I think the homework is first making sure the clinical standard is applicable to the US, and since we are talking about US FDA, or to any other regulatory agency. Those are the things, and really sponsors need to make sure that that the data that would be generated from foreign countries would support their marketing application in the future, taking into account the clinical standards. That's what I would say, Kim.
Thanks, Casa. And Anne, we'll shift gears slightly to you. There's a couple of questions here about other types of requirements. So what trials are required to be registered in the clinicaltrials.gov databank?
Wow, that's such a good question, and one where I think if we think about clinicaltrials.gov and trials transparency, where the agency is really committed to promoting transparency and will continue to advance our compliance activities related to the clinicaltrials.gov database. So the requirements apply. The trials that are required, that are subject to the requirements, are applicable clinical trials. And the section 801 of FDA, including its implementing regulations at 42 CFR Part 11, or the other Part 11, as referenced in my presentation, specifies requirements for submitting registration and results information for these applicable clinical trials. So an applicable to clinical trial generally meets all of the following criteria. It's interventional, it's evaluating at least one drug, biological, or device product or combination thereof regulated by FDA. It's other than a phase 1 trial of a drug or biological product, or is other than a small clinical trial to determine the feasibility of a device. And it also has to meet one of the following criteria. At least one study facility is located in the US or a US territory, or it's conducted under an IND or IDE, or it involves a drug, biologic, or device product that's manufactured in and exported from the US or a US territory for study in another country. So I think that's the kind of definition of an applicable clinical trial. And those who have to register those applicable clinical trials and report summary results when required are known as the responsible party. And under clinicaltrials.gov, this is, under the regulations, this is the sponsor of the trial unless and until a principal investigator has been designated as the responsible party. So if a study is conducted under an IND or under an IDE, the IND or IDE sponsor is generally the responsible party. If an applicable clinical trial is not conducted under an IND or IDE, the responsible party would be the single person or entity who initiates the trial and has authority and control over the trial. So again, really excellent question. Thank you for asking.
Yeah, great. A lot to unpack there. Another question on requirements. Is a written delegation of authority log a requirement?
It is another good question. So our regulations are silent on the delegation of authority log. Where you will see delegation of authority logs referenced is in guidance. So for example, in the ICH E6 R3 draft guideline in section 2.3.3, the statement that the investigator should ensure a record is maintained of the persons and parties to whom the investigator has delegated significant trial-related activities. You'll also see that referenced in guidance, FDA's own guidance on investigator responsibilities, but it is not a requirement that's within our regulations. Thanks.
Thank you. And one more for you while I have you. On slide 7, you said, quote, transfer responsibility, which does not align with my understanding. GCP is clear that responsibility remains with the sponsor. Do you mean transfer of work activities?
So that's a good question. So I was referencing our regulations, our IND regulations under subpart D, the responsibilities of sponsors investigators, actually talks about in 31252 the transfer of obligations from a sponsor to a contract research organization. So a sponsor may transfer responsibility for any of all of the obligations set forth in this part to a contract research organization. So those things need to be transferred in writing. I think what the questioner is referencing is that you can transfer responsibility for an obligation, but at the end of the day, a sponsor is accountable for the overall quality of the trial and for the information submitted to the agency. So you still need to have appropriate oversight of the responsibilities that you've transferred. Thank you for giving me the opportunity to clarify.
That's great. Great questions. Stephanie, a couple clarifications on terminology from your talk. The first is, what specific certification does, quote, laboratory certification refer to? Please provide examples.
Hi, thank you for the opportunity to clarify. The certification I was referring to is a certification from an organization that certifies the lab is able to do its job, receive human samples for testing, and on that end. So a common certification in the US is the CLIA certificate, Clinical Laboratory Improvements Amendment Certificate. Labs have to be certified with CLIA so that they can receive payments from Medicare or Medicaid. So that's a common one in the US.
Great, thank you. And then another terminology question. One of your slides referenced, quote, data flow. What is that referring to?
Hi, yeah, thank you again for that question. So I was referring to how the data that you generate at your site gets transmitted to the sponsor. So, you're recording the data initially. It might be on paper, it might be electronically, and how does that get to the sponsor intact, or are there any issues with data integrity or security? So if it was on paper that you originally recorded information, like on a study worksheet or something, how was that transferred to the sponsor? Did a research coordinator enter it into an electronic database? And, were password protections in place that only specific people could do that? And were there any issues with alteration and things like that? Sometimes assessments are made electronically through tablets or other devices. And so and then it's transmitted automatically to the sponsor. So things we might look at there are things like audit trails, kind of to see who entered the data, that specific people were entering the data, and that logins and passwords weren't being shared. And then when it was transmitted to the sponsor. So basically, how everything went from you all and into the company.
Great, thank you. And then a more general question is interested in understanding how likely it is to have a site visit and inspection.
Yeah, that's a great question. Things that we look at for a site inspection, in particular for those related to a marketing application, we look at the size of the site itself, how many subjects were enrolled, how well your site contributed to total study efficacy. Was it a outstanding trial site, and so was a big contributor to study efficacy? Was your study site an outlier for adverse events? And it might not just be that you had a lot of adverse events and protocol deviations. That could just be you doing a great job in terms of reporting those. It might be that it looks funny that you didn't have any adverse events or protocol deviations. And so we might want to look at those. If you've had a previous inspection history that, we might want to touch on some of the things that were found at that previous inspection history. We might be considering going back. And as I kind of touched on in a previous response, the sponsor, as part of the submission, informs us of any issues at particular sites. And so if there was any concerns by the sponsor about data integrity, subject safety, and those sorts of things, those are some things that we might look at for choosing your site. We have a specific tool that helps us, so it's not kind of willy-nilly. It's not a lottery. It's we take all those factors into consideration.
Great, thank you. Casa, Anne got some questions earlier about the Form 1572, and we have a question from more of an international perspective. The question is, as far as I know, investigators in the European Union are not allowed to sign Form 1572. What should a U.S. sponsor do when conducting a trial in the European Union for a product under IND?
Not the entire European Union, and in the European Union, some countries have local regulatory requirement that asks their clinical investigators not to sign 1572. I think Anne addressed that, and there is a mechanism for that. If the clinical investigator had signed an IND or went to sign an IND, that would be considered. Clinical investigators may be able to conduct a study under IND after having the waiver requirement made, and they can ask to conduct a study under IND and gate the waiver. But during my talk also I mentioned outside US, clinical trials in support of marketing application can be conducted under IND, or there is no requirement to conduct clinical study under IND, and they can be conducted outside IND. And however, if clinical trial is conducted under an IND, the requirements that people have just discussed must be made. And during inspection, our inspectors take into account some of the local regulatory requirement that the clinical investigators have to meet. Therefore, if there is like a requirement, let's say Germany may ask the clinical investigators not to sign 1572, we do take that into account. We recognize, and we do say also they need to meet in ICH those A6, R3, and before local regulatory requirement, meeting local regulatory requirement is one criteria. Therefore, the clinical investigator should be proactive and take this into account. And if there is a difficulty to sign 1572 because of local regulatory requirement, they need to work with their sponsor to have the waiver recognition, or they should conduct the study outside IND. Kim?
Great, thank you, Casa. Anne, are there any guidance documents specifically for risk-based auditing, risk-based monitoring?
Oh, I love this topic. Because I really am an advocate for risk proportionate approaches to oversight. We actually have quite a few guidance documents that touch on risk-based monitoring or risk-based quality management. So from the specific to risk-based monitoring, there is a final guidance on oversight of clinical investigations, a risk-based approach to monitoring that came out in 2013. There was a more recent 2019 guidance that on risk-based approach to monitoring of clinical investigations. That is a Q&A document. And then, of course, there is the ICH E6 R3 that discusses risk-based approaches and kind of risk proportionality in terms of oversight. So I think there is, if you're interested, the FDA does have a website that we have all of our clinical trials related guidance documents that if you just search for clinical trials guidance FDA, you should be able to find it pretty quickly because it's a very useful resource where you can kind of filter down to guidances on the specific topic that might be of interest to you. Thanks.
Great, thanks, Anne. And Casa, a follow-up question for you that's related to Anne's question. From an inspection perspective, what are the key aspects you would look for when reviewing risk-based quality approaches?
Thank you, Kim, and that's a great question as well. And really, the risk-based approach, first of all, it's a dynamic process, and it starts, and it needs to occur throughout the lifespan of the drug development, and therefore we would like to see the pre-specification and the way how people have determined what are important, what are critical from data reliability perspective, from process perspective, impacting the reliability of data, from patient safety perspective. Based on the pre-specified criteria, have people followed? Really, the key is, I think Anne pointed out, what we care during inspection is to make sure that the things that matter most have been properly overseen, and those are the things that we see in connection with our regulatory requirement. And perhaps, I mean, as most people here have seen, our inspector focus, for example, on informed consent, ethical development of the product. Has that happened throughout the lifespan of the drug development? And we would care about primary endpoints that are critical for labeling of a product, and also to make a claim about the product efficacy. And of course, regulatory expectations would be seen. But the bottom line is, how were those risks identified, and have people followed the appropriate steps once those risks had been identified during the study? Those kind of things are what we care about during inspection. Kim?
Thank you. And an inspection-related question for Stephanie this time. When sponsors, not CROs, undergo inspections, what aspects are most crucial for inspection?
Hi, yeah, thanks for that question. Just like for clinical investigators, clinical inspections and what we look for are really tied to the entity's regulatory responsibilities. So I know Anne gave a really nice presentation on the regulatory responsibilities for clinical investigators. Sponsors themselves have their own regulatory responsibilities, and so the inspection is focused on that aspect. And so some things we look at are the how they selected and trained and kind of monitored the clinical investigators and the actions at the clinical sites. You know, if there were issues at the clinical sites, you know, what corrective actions did they take? And if they weren't corrected, were sites terminated? So kind of their overall oversight of clinical investigators. Likewise, their oversight and management of contract research organizations and third-party vendors. We look at that, how they chose their study monitors, their monitoring procedures and activities, and kind of taking I talked about data flow from the site to the sponsor level. You know, what was the data flow from the sponsor to FDA? What kind of manipulations to the data they did? Their responsibilities for adverse event reporting. We look at that, and their record retention and things like that. So it's really tied to their regulatory responsibilities and anything of particular interest to the particular studies we're looking at, or what the review division is also interested in focusing on.
Thank you. Another question for you, Stephanie. Does FDA inspect against clinical investigator, sponsor, or CRO standard operating procedures?
Hi, great question. Yeah, we my talk focused mainly on protocol compliance and kind of using the protocol as kind of the guidebook of what we're looking for in inspections. But we do look at standard operating procedures that the site might have, that the contract research organization may have, and the sponsor may have. It kind of just helps round out our picture of how the study was conducted, how it was managed and reported. So it gives us good ideas of why data was recorded in a certain way, why it was reported in a certain way in the study report, why certain things weren't reported to FDA. It kind of gives us a global picture of the study conduct beyond the protocol and study documents.
Thank you. And another one for you. Sorry to keep you on the hot seat for so long, but clearly a lot of interest in this topic area. How long should a site store the clinical study samples at facility and also investigational products? I'm not sure that's worded quite right, but hopefully you know what they're getting at.
Yeah, one of the earlier questions had talked about study records. So yeah, you're probably wondering what to do with all the other things at your site. So if, in terms of, an investigational product or samples, usually some of that of what to do, because you are responsible for that as part of your regulatory responsibilities, that might be outlined in the protocol or a pharmacy manual about how to dispose them or send them back to the sponsor. So definitely look in those areas of the protocol or manual. And otherwise, I would recommend just checking with the sponsor or your study monitor, who might be more accessible.
Great, thank you. And last one for you for this part, I promise. Does inspectional approach defined in CP 7348.811 apply to a sponsor, or is there an alternate CP the FDA applies?
Yeah, great question. And thanks to the person who's really paying attention to that compliance manual. Yes, there's a different compliance manual for sponsors and contract research organizations. And actually, it also includes sponsored investigators. So it might apply to some of those of you out there. And so that number is the same number, just with the 0 at the end. So 7348.810, and that's also found on the website as well. Some of the things I talked about and related to an earlier question about, what things we look for at a sponsor inspection are included in that manual. So I kind of summarized a little bit about what you might see if you actually went to that and looked at the manual.
Thank you, Stephanie. One more question for you, Anne. You had addressed the informed consent process earlier and answered some questions related to informed consent. This one is about the assent process. Can you please speak to the assent process for pediatric subjects?
Sure, I would be happy to. So our regulations at 21 CFR Part 50, Subpart D, provide additional safeguards for children enrolled in clinical investigations. So if a child is going to be enrolled in a trial, parental or guardian permission must be obtained and documented. And when appropriate, the child's assent must also be obtained. So this is something, assent essentially means a child's affirmative agreement to take part in a clinical investigation, not just that they failed to object. So child's assent when appropriate, and that's something the IRB needs to consider, and parental or guardian permission taken together really meet the ethical requirement to obtain informed consent. And I think for those who are interested in ethical aspects of pediatric clinical investigations, my colleagues in the Office of Pediatric Therapeutics published a draft guidance in September of 2022 on ethical considerations for clinical investigations of medical products involving children that I think is an excellent read. I would also direct you, which I don't think I mentioned earlier, that just a few months ago, we published a final informed consent guidance that updates an older draft guidance and has a wealth of information, both with just general information about the different aspects of our consent regulations, all the way through a series of questions and answers about various scenarios. So there's a lot of resources available for those who have questions about informed consent. So thank you.
Thank you. And I have a couple of questions here about global studies, not directed to an individual, but I assume, Casa, that this is for you. In a global study, is it okay if different country sites use different instruments or platforms for data production or analysis, as long as they have validated methods?
Thank you, Kim. Yes, and it would be okay. It is very important to have a discussion with the sponsor, and I think we may be talking about a sponsor or CRO responsibility, and this is just like having paper based source records versus using an electronic platform. Both are fine, and the question is, would those instruments or tools would be fit for purpose to generate and to capture the information? That needs to be taken into account. But in terms of generating the data, I think the part 11 is one of the things that was mentioned here. People need to take into account if they use electronic kind of records. They need to meet that expectation, but using a different platform is absolutely fine if the tools are properly validated and if they are compliant with the expectations. Kim?
Thank you, Casa. And one more for you. In the efforts to diversify participation in clinical trials, what are the sponsor's responsibilities in conducting trials, for example, in Africa or South America?
This is a great question, actually. It would be that, I mean, first and foremost, it would be in the interest of the sponsor to diversify the patients because the study data would be applicable across the globe if they have diversity. Of course, from patient perspective and public health perspective, it would have also a huge value. And there are various things that may have been mentioned during discussion yesterday, the different kind of approach, including decentralized feature and considering real-world data and trying to make the studies like patient-centric through the decentralized feature. All this could enable diversifying the patient population that would participate in clinical trials that would have not been able to participate. And since regions have been mentioned, it is very critical for the sponsors to include patients across the globe, taking into account what the product is going to be developed for. And therefore, the sponsor responsibility is really identifying the benefit of diversity and also implementing that to happen. And there may be instances where sponsors may not be able to diversify. Let's say there is a tropical illness that may not occur in some other countries. And those things are very straightforward, but diversifying patient population or study participants is very critical, both for a sponsor as well as for public health. Kim?
Absolutely, and a lot of work ongoing in that area currently to try to make sure our trials represent the patients who are out there using these products. So on that note, I would like to thank all of the presenters for both excellent presentations and for fielding a large number of really good questions during the question and answer period, and also thank the audience for putting these questions into the chat to allow us to have additional discussions of these issues. And with that, I would just like to wrap up this session and hand it over to Leonard for some closing remarks on the course.
Thank you very much, Kim, and thanks very much to the presenters, to Anne, Stephanie, and to Casa. Well, that brings us to the end of this course. We've covered a lot of ground with regard to clinical trials, the clinical, the scientific, and the regulatory aspects that are important. And we very much hope that this has been helpful to you as you go ahead with a very important contribution you're making to the health of society. Obviously, it's not possible to cover all aspects of clinical trials, and we would welcome your feedback to help us as we prepare for the course next year. It remains for me to thank all my colleagues involved in the course. It's been a big production. A particular vote of thanks to Brenda Stoddart and SBIA for basically supporting the course, to Ray Ford for all his help in keeping us on track with housekeeping, to Jeff Kelly for his excellent and competent handling of the webinar and dealing with all the storms that happen behind the scenes in such a competent and quiet way. Thanks to Millie Double from my office for all the help with the administration and organization, and to Kim Smith, who's been a great co-moderator and has helped us significantly with developing the context of the content of the course. I want to thank all speakers, and finally a particular vote of thanks to our audience from all over the world for your dynamic participation. So from us at FDA, our sincere thanks to you all for taking part in the course.
Thank you, Dr. Sachs. This is Ray Ford. Just a few closing reminders for our audience. In addition to the speaker evaluation, we have our final evaluation. It'll be available later on this evening from our one-stop shop link. This is the final survey is the mechanism by which you'll receive the certificate of attendance, which will support CE credits for RAP, SOCR, SQA, and ACRP. Be sure to download the certificate immediately upon completing the survey, otherwise this opportunity may be lost. Even if you don't want to need the certificate, please take a few minutes to complete the conference evaluation, as your feedback will help guide the design of our future event. Note that the evaluation and are only available for 2 weeks, and that takes us to December the 21st. Also, to summarize, for our physicians, pharmacists, and nurses claiming the continuing education credits, CEs are available if you're registered and attend in real time. SBIA will issue the CE claim code and instruction to all attendees later tonight, so be sure to complete the CE session evaluation also on or before December 21st. I apologize in advance, but we are unable to make exceptions to the deadline. Attendees have 14 days from the last day of the activity to log in, complete the required CE evaluations, and attest to attendance to claim credit. Tonight's email will have both the claim code and instructions together with the survey link. As a final reminder, all sessions are being recorded. It'll take us a week or so to process recordings, but you'll soon be able to access them through SBI's webpage, and that's fda.gov/CEDAR. On behalf of CEDAR Small Business and Industry Assistance, we hope that you enjoyed today's course presentations and found them helpful. Look forward to your presence at our next SBIA event. This concludes the FDA Clinical Investigation Training Course 2023.